Okay, everybody, my name is Sherry Flogi. I'm so excited to have this opportunity to share my love of marine viruses with you and also hopefully to introduce a fascinating global biogeochemistry. Have this session today and also thank my co-chairs, Jessica Labonte. She wasn't able to join us in person, she is on Zoom. Jeff Bowman, who's here in the audience. I'm going to begin with a brief introduction and then we have some goals for this session. So the first goal is to foster connections between the marine virology community and the OCB. All right, hopefully you can hear me better now. Okay. So foster connections between the marine virology community and the OCB. So what I've heard from a lot of people is that we know there are a lot of viruses out there. We know they're doing something, but we don't quite know what they're doing. And so as marine virologists, we often pitch our proposals and our setup frame our publications from the context of viruses are important in carbon cycling. But I think connections between the two communities are under how viruses are mediating that. And then because many of you said, we know viruses are out there, but we're not quite sure what they're doing, I'm going to begin our session with a little bit of an unusual talk, which is two graduate students, Kate Thompson and Jordan Walker, are going to co-present an introduction to viruses, marine viruses, bring everybody up to speed. And then we'll have we want to summarize the current state of knowledge in the field of marine viruses and carbon cycling. We'll be doing this through Kate and Jordan's talk, and then also through our speakers. And then last, all of our speakers have been asked to highlight knowledge gaps and emerging tools to overcome such gaps. Okay, so why now? Why marine viruses now? There's been a number of commentary and perspective pieces published in the last few years highlighting the role of viruses in biogeochemical cycles and the new tools available to tackle this problem. It's been around for some time. This is a illustration from and Vardy's showing some of these new tools and some of the ways that we can try to understand how viruses are carbon in the ecosystem. There's some other additional papers I want to highlight here, and one that was bringing in explicitly modeling viruses in models. What I noticed about a lot of these reviews is that they are published in papers that are targeted, or published in journals that are targeted to microbiologists, so microbiology journals, virology journals, et cetera. And so there is some question of whether these papers are being seen by the larger carbon cycling community. And so first, we have a great lineup of speakers. First is, again, Kate Thompson and Jordan Walker, who is going to give you a primer on viruses in marine ecosystems. We'll then have Cynthia Silvero, who's joining us via Zoom to talk about who infects whom, identifying productive viruses by combining sequencing with abundance data. Christina Mojica will be speaking about quantifying the viral shunt in the North Atlantic Ocean. And then myself, I'm going to state for the record, I did not invite myself to speak. This was the idea of Heather and the other organizers. But I'll be moving from ecosystem scale to the scale of single microbe interactions at the time scales of seconds to minutes, so understanding how those interactions can influence larger scale ecosystem processes. We'll have a break, and then we'll move into the modeling component of the session. So David Talmy will be talking about empirically modeling carbon flow through marine viruses and microzooplankton grazers. Daniel Murator will be talking about generalizing viral elemental composition and infection as trophic transfer. And then we'll have a panel discussion I invite you all to participate in. And then hopefully by the end of the session, you will discover a newfound love of marine viruses and you will want to talk more with us. You can join us for lunch at tables 1 and 2 and also check out the OCB webinars. We had a couple webinars leading up to this meeting and they are posted online. Okay, I'm going to turn it over to our first speakers now. Thank you.
Thank you for having us. Thank you, Sherry. My name is Jordan Walker. I'm from Galveston. As she said, I'm a PhD student.
My name is Kate Thompson. I'm also a PhD student at Wake Forest University in North Carolina.
So when we were asked to sort of give you a primer to viruses in marine ecosystems, we started with this figure from Frontiers in Young Minds. So it's a paper that that has children or high school students that help come up with the figures and the language that goes into these. So we have a very simplified version of the marine microbial food web with a focus on phytoplankton and bacteria and the microbial loop of the consumption of that, the organic matter produced by phytoplankton by that bacteria. We also have viruses in that top right corner, and our goal here is to sort of describe what those viruses are and what they're doing. So we had 4 main questions we wanted to answer for what are viruses, what do viruses do, why are viruses important, and how can we assess the impact of viruses. Just to jump right into it, viruses are obligate parasites. They have to find a host cell to infect, which happens up here at the entry stage. And then after entry, we can start into the viral production life cycle. The most known viruses are these lytic ones in blue, where production ends in the death of the host and the release of organic matter from the host into the dissolved in particulate phases. The next well-studied virus out there is the lysogenic or temperate ones. And these actually go into a stage where they're not productive and they're integrated into their host DNA. Something happens in the environment that will induce them into a productive stage at some point. And then the ones that we know the least about are the chronic ones. We know the least about them because we don't actually see death of cells happening. So the production of viruses, we can't really see that happening in the lab. So I showed you a very simple picture of sort of what viruses do and how that affects geochemical cycling, but I want you to keep in mind this is a very complex process with a lot of different steps taking place throughout time. And viruses do all these steps in different manners and different ways. So how we get from one step to the other is a very complex process out in the ecosystem. As far as the total diversity of viruses, I wanted to sort of take you through a history of how we've done taxonomy of viruses, because this does a really good job of summarizing the diversity of viruses. So originally, we separated viruses into 3 major groups: viruses that infect bacteria or phaginae, viruses that infect plants or phytophaginae, and viruses that infect animals or zoophaginae. We now know there's also viruses that infect other viruses, so not quite complete there, but a good way of describing the diversity of viruses. We then started describing viruses based on the Baltimore classification, and this was based on what types of genetic material the virus hosted. So just like us, we can have double-stranded DNA viruses, but we also have single-stranded DNA viruses and viruses that use RNA instead of DNA, both double-stranded and single-stranded as well. As we moved on from there, we actually got a whole committee for taxonomy of viruses. And as we got transmission electron microscopy images of those viruses, we started to realize that the morphology of viruses is very diverse, and that's how we started classifying them. So you can see your typical marine viruses, Narcotiviralis over here with their nice head and tail that a lot of us are used to, as well as things like our Mimi viruses that are giant viruses and on the order of magnitude of cell sizes. We've had one more revolution in viral taxonomy, which came because of shotgun metagenomics, or sequencing of total DNA from the environment. And this has allowed us to almost double the size of viruses that we've been able to describe. And we do this based off of the nucleotide identity of those viruses, as well as the gene content. So the first metagenomic studies of viruses really happened in the early 2010s. And as you can see, the number of species we've described since then has exploded. So answering that first question, what are viruses? They're obligate parasites infecting all known life. There's a huge diversity of them, from the types of genetic material they use, the gene content, morphologies, and lifestyles that they utilize. Now, Kate's going to talk about what they do.
Thank you. Okay, so as obligate parasites, we know that viruses cause a lot of mortality in the ocean. And lytic viruses, that happens on the scale from hours to days. And with lysogenic viruses, that can persist across generations. It's estimated that about 20% of marine microbes are infected on a daily basis. And Viral lysis has been estimated to be responsible for about 50% of phytoplankton carbon loss in the ocean. Additionally, some of the main mortality factors of cells in the ocean fall under grazing and viral lysis. The study on the right here is a study from 2022 by Ike et al., showing that viruses have a lower rate of infection compared to grazing or lysis. But astonishingly, on the right, you can see that despite that, the mean carbon loss is about equal. So this is just one piece of evidence suggesting that viruses play a really large role in nutrient and carbon cycling in the ocean. So looking at this food web dynamics, so in our field of marine virology, we kind of classify viruses' roles under two different hypotheses. The first one being the viral shunt, which is the viral lysis contribution to the DOM and POM pools, fueling heterotrophic bacteria and phytoplankton growth, shunting that organic matter away from higher trophic levels into the microbial loop, which we've heard a little bit about thus far. And then alternatively, or not alternatively, but there's the viral shuttle, which is the virus-induced aggregation of cells that leads to larger, heavier particles sinking carbon with depth at a quicker rate. But we know viruses are not only causing mortality of cells, but actually altering those cells physiologically. So a lot of marine viruses encode what we call auxiliary metabolic genes, or AMGs, that are thought to actually alter host metabolism and redirect those pathways towards virion production. With lytic viruses, which is what this figure is depicting, a lot of those auxiliary metabolic genes are more related to those metabolic shifts, whereas with lysogenic viruses, those AMGs are related more to the longevity of the host, so keeping the host alive. So that's the AMGs in this figure are highlighted in red. So I guess related to this, not only do viruses alter the physiology of the host, but we also know that they alter the biochemistry. There's evidence of metabolic pathways shifts from fatty acids or towards fatty acids and nucleotide biosynthesis during infection. So here on the right is a study by Ankra et al., 2014, not good with laser pointers, showing the metabolite signature of a virally infected bacteria by roseo-phages. And we see this across a lot of different systems, which we'll hear a little bit more about today. So what do viruses do? We know they cause cell lysis as obligate parasites. We've learned a little bit about the shunt versus shuttle dynamics. They hijack hosts and alter their metabolism, and ultimately they alter host biochemistry. Now I'm going to send it back over to Jordan, try not to go back and forth too much, on why viruses are important.
Yeah, so we didn't always quite understand how important viruses are. The first evidence of this came in 1989 with this great short paper where we got the first transmission electron microscopy of seawater looking for viruses, and we were able to count something like 250 million viruses per milliliter seawater. Most of these are the bacteriophages, which you've seen so far here in like panel C and B, where they're infecting mostly bacteria in the ocean. They're not infecting humans or animals or plants. And since then, we've done a lot of counting of viruses, and what we found is that 250 million number is pretty accurate. So globally, we think about viruses being somewhere in the range of 10 to the 6 to 10 to the 9 viruses per milliliter seawater. But this depends on where we look. So in more productive regions like coastal and estuaries, we tend to find them more in that 10 to the 7 number, whereas the open ocean, you find them about an order of magnitude lower. They also follow their host range, so most of those being bacteriophages are going to follow prokaryotic abundances. What we tend to see is this VPR mean, or this virus to prokaryote ratio, tends to be 1 order of magnitude higher for the viruses compared to their bacteria. Recent evidence is sort of suggesting that this is actually more complex and really depends on the lifestyle of the host and the virus, how they're interacting together. So we know they're super abundant. We can scale up those numbers and find something like 4 times 10 to the 30 viruses total in the world's oceans. And this makes them by far the most abundant biological entity, as we can see in this bottom right panel pie chart. But because of their small size, mostly in the DOM fraction of the phages up here, the top panel, the biomass of them, for them, only accounts for about 5% of the total biomass in the ocean. Their sizes do vary wildly, so we can go as small as microviridae, where we're getting 15 nanometers. These are single-stranded DNA viruses, or we can get as large as this pithovirus sibiricum at 1.5 microns, bigger than some bacteria out in the ocean. On average, though, what we're looking at for pelagic samples is about a 50 nanometer virus. Just to kind of give you an idea of how big viruses can get, this is a picture of pithovirus with scales on panels C and D. You can see quite how big it is. And then if you're really interested and want to like scare your children, you can actually buy a fuzzy version of this with weird zombie eyes. So we know they're everywhere, but can we say something about their actual biogeography? So the most comprehensive data set we have for this comes from Terra Oceans. It was a 4-year sampling effort. We were able to make 43 viromes, and from that we were able to describe something like 6,000 different viral populations. What we really found here is that viruses, no matter where we find them, so the original station is where we found that virus from, we typically find them in more than one sampling location. So I think on average, they found them in 4 to 5 different sampling locations, but up to 25 different sampling locations. So indicating that viruses are globally distributed and they're not only in one range. The bottom panel here is showing their relative abundances. So even though they may be globally distributed and found in multiple areas of the ocean, they are more abundant at certain locations at specific times. As far as the time goes, I wanted to talk a little bit about that. The most comprehensive data set we have for that comes from a spot in right outside of LA. They had 5 years of monthly sampling doing viromes. They were able to identify about 20,000 viral populations from this. And what they did here was they compared the similarity of the community on the y-axis, and then on the x-axis, this is the amount of time between each sample that they're comparing. So what we see here is you have peaks in the similarity at every 12 months, indicating seasonality to the viral communities. And then every 6 months, you have the valleys of these sine waves, indicating the most difference between those. One other thing to note here is there's a slight negative correlation as we go along with this, indicating that these viruses are actually evolving with their host and changing through time. They're stable with their host because of that evolution with them. So why are viruses important? They're the most abundant biological entities. They're found globally, and they do have these long-term stable interactions with their hosts that we've identified.
Okay, so now assessing the integration of viruses into biogeochemical models. That is the main goal of our session here today. So many models that have begun to incorporate viruses as components have focused on viral mortality based on host density dependency. But we know that viruses are doing more than just killing their hosts. So there's evidence of viruses playing a large role in the carbon and nutrient cycling, as we've gone over a little bit today, and you'll certainly hear more about this morning. And we know that we really need to begin incorporating these viruses into complex biogeochemical models to understand those virus-host dynamics at a relevant ecosystem level. But there's a reason we haven't done this yet. So there are lots of challenges in incorporating these viruses, and they are very complex and variable. And I have a list here of some of the traits of viruses that kind of tap into this, starting with morphology, which we've talked a little bit about. So there's bacteriophages that have icosahedral heads, spherical viruses, helical viruses, tailed, non-tailed, enveloped, non-enveloped, a plethora of different morphologies. And then in turn, that can affect the host specificity and range. So do we really have a grasp as to who the generalists versus the specialists are in the marine environment? And related to that, there's the latent period length, which is the period before the cell's lies under infection. And we know that there's a lot of variability in that length, even amongst viruses that are closely related. Additionally, there's burst size, which is the number of particles that are released per cell. And that can range from 6 to 300 particles per cell, depending on the virus. There's a lot of variability in the production per each virus. And then the fitness, do we know what percentage of those virions that are released? Do we know what percentage of them are infectious? As Jordan has touched on, we know there's a lot of variability and complexity with the different lifestyles, so lytic versus lysogenic viruses and others. Genome types, right? So there's single-stranded RNA, single-stranded DNA, double-stranded RNA, double-stranded DNA, and that the actual genome size of these viruses can range anywhere from 4 to 630 kb. So there's a lot of variability and complexity as kind of the goal of this talk. I hope we've communicated that to you. So these are just some of the challenges that we have to keep in mind with incorporating viruses into these models. So we've talked a little bit about integration of viruses into the models and defining different traits and challenges. And we hope you keep in mind as you listen to the remainder of the talks in the session, you know, we want to facilitate conversation and come together as marine biologists, biochemists, and modelers, and everybody in this room to sort of come up with steps moving forward. So with that, thank you. And I'm not sure if we have a couple questions, or are we moving it on to questions? Thank you.
Anyone have a burning virology question they want to ask? No question is too simple.
Are viruses alive?
So typically, how long do viruses live outside the cell? How long can they survive on average?
It's a great question. I think that varies a lot virus to virus. We know that a lot of bacteriophages have really slow decay rates. But, you know, depending on the density, they're bumping into cells left and right. So my answer to that is that it varies extremely.
Not only that, but where we find the virus also sort of depends on how long it stays active, right? So UV degradation is something you need to consider, and where it is in the water column is important. I recently got into like hypersaline viruses, and one thing that they talk about there is the production of different glucose molecules that can help protect the virus and keep it stable over time. So yeah, it really depends.
Hi, thanks very much for the presentation. As a non-expert, I really appreciate it. This sort of a primer on what's going on, and I'm asking a question from the perspective of a biochemical modeler who might want to, you know, include more realism into our models. And on the one hand, you know, all the diversity you presented, all these different groups is kind of, you know, scary. It almost stymies the ability to like feel like we could make any progress on this, given this diversity. But then, you know, you sort of think about it, and you could say the same thing about phytoplankton, right? There's many different groups of phytoplankton and many different types of phytoplankton. And we've made a lot of progress in models by thinking about functional types. So we've sort of tried to classify the inclusion of phytoplankton in models by their biochemical role or by their size. I just wondered what your views were as to whether that was a tractable solution to sort of compartmentalize the diversity a little bit.
Yeah, I think it is. We've already sort of begun to do that. There's groups like Joshua Weiss's and David will talk a little bit later on that, but trying to compartmentalize them into who they infect or how that infection process takes place. So that's been the most progress that I think we've made so far with those viral models. But yeah, you know, doing all of them everywhere is always going to be problematic because of the immense diversity and the fact that they're constantly evolving.
Okay, will you join me in thanking the speakers again, and we'll move on to Cynthia. Okay, our next speaker is Cynthia Silvera. She's joining us via Zoom because she unfortunately couldn't be here today. She's assistant professor at the University of Miami, and she's going to talk about some progress she's made on who infects whom, which has been this problem in marine virology, somewhat intractable problem for a long time.
Excuse me. Good morning, everyone. And I'm really sorry for not being there in person, but I have COVID, so you really don't want me to be there around you. And today, I also really want to thank the organizers for accommodating my presentation online. I'm really excited to share this work with you. It's actually the first time I'm sharing these results with the community. They haven't been published yet. Hopefully they will be soon. So we're really interested in this question of who infects whom. And we're going to be focusing on this talk on a subset of all this diversity of viruses that was just introduced to you. So these are going to be the double-stranded DNA viruses that infect bacteria. So I'm just going to be calling them phages for the most part in this talk. And even though they are a small subset of this diverse world of viruses, they actually account for the great majority of viruses that we find both in the plankton and in sediments in marine environment. And when I say great majority, I'm talking about 95, 99% of the viral particles that we can identify. So they look like this in this picture. So they have this capsid or the head where they store their genetic information, and this tail that they use to inject their genetic material in the host. And they are known to kill bacterial cells. So they enter the host cell and they replicate. And to get out, they have to blow up the bacterial cell. So this is what they are most known for. And there are estimates that 20 to 50% of the new bacterial production in the plankton in the oceans every day is killed by these viruses. But of course, given the great diversity of viruses and bacteria that we know is out there, you would expect to find a lot of nuance in how these virus-bacteria interactions is actually taking place. So just a very brief example here, which is something that I have been studying throughout my career, is this duality between these two different types of infection strategies that these double-stranded DNA phages can choose. So in the left side, you have the lytic strategy, which is the one that we just talked about. The virus goes in, blows up the cell, releases viral particles. And then the right side, you have the temperate viruses. So those are the ones that have the option to integrate their genome into the bacterial genome and stay there in a long-term interaction. So the bacteria keeps growing, that virus gets replicated alongside the bacterial genomic information. So this is a completely different type of ecological interaction that is actually a lot more common than we thought just a few years back. And there are other types of infection strategies that was mentioned earlier today, like chronic infections. They seem to be a lot less common in marine environments. But there are all these possibilities. And even within the lytic infections, you could imagine that viruses with different burst sizes, which is the number of viral particles produced at each infection cycle, or the time that it takes to lyse the cell, or the decay rate of these viruses in the environment will be different, right? So for the purposes of this workshop, I think asking this question of who infects whom is very relevant, right? Because we want to start getting into these life history traits and start grouping viruses by who they infect, how fast they infect, how many viral particles they produce, how long they stay in the environment, and so on. The problem is that the technology for addressing this question has been very limited. So on the left side, what you see here is a petri dish where you have the bacteria growing in the background. So that's your sort of whitish part. And then those clear spots are phage plaques. So that's where the phage kills the bacteria. So you can go in the field and you can collect seawater and you can grow bacteria and viruses like this and find who infects whom. But of course, you're limited to who you can grow, which is a very, very small fraction of the bacterial community and the viral community too, of course. On the right side, you have bioinformatics approaches, right? And they're really great because they're very large scale, but their confidence is much lower. So typically, we can only make predictions at very broad taxonomic levels, which ends up being not as informative for us, and or depending on the question, of course. And what happens here is also that the main tool that we use for this bioinformatic host prediction are CRISPR spacers, the same CRISPR that we use for gene editing nowadays. But marine bacteria tend to have very few CRISPR spacers. So we are left with a few options here. Some of the other technologies that I didn't put here are single cell sequencing, which is really great because it combines a little bit of both of these advantages of these two methods, but it's really not high throughput yet. And I would like to see that technology developing better to make it cheaper, more high throughput for us. But what we decided to do for this project was to use this other technology called proximity ligation or Hi-C. So, excuse me, in this technology, what we do is we cross-link DNA that is inside a cell before we sequence that DNA in a metagenome. So this technology has been around for a long time and was used for studying the three-dimensional structure of chromatin in eukaryotes. And then it was adapted to studying plasmids and other mobile genetic elements in bacteria. And until now, it was only limited to very high density samples. And then in this study, we were able to adapt this technology to low density seawater samples. So what you see here on the right side of this graphic is you have a bacteriophage replicating inside a bacterial cell. So you have both the cell genome and the viral genome in close proximity inside the cell, and you can cross-link these molecules. And when you sequence them, you can tell which virus was replicating inside which bacteria. And this technology is really cool because it will give us information on viruses that are replicating lytically. It will also give us information about viruses that are integrated. It will give us information about viruses that are establishing chronic infections, so they are not integrated, but they are also not killing the cell. So any viral genome that is inside the bacterial cell. And I want to highlight here my student, Natasha Varona, that was the person that worked with me on this project. She really led the development of the method, both bench and bioinformatics for this. And Natasha will be looking for a postdoc next year. So I'm just doing a shameless advertisement here of my student. And so we combined this Hi-C method with the bioinformatics approach that were available to us. So we did search for CRISPR spacers. So that's the figure that you see in the left, where we match sequences in the viral genomes with sequences in the CRISPR arrays of bacteria. And we had, of course, very limited success with this method, but we used it nonetheless. And we also searched for those integrated viral genomes that I mentioned to you. And we did this in these coral reef waters around the island of Curacao and the South Caribbean Sea. And so this is a coral reef environment, so it's not quite your open oligotrophic ocean, but these waters are still pretty oligotrophic, and the bacterial densities here are around low 10 to the 6 cells per ml. And so I think that this is a really nice model system here. Our dominant bacteria in these waters are still your Prochlorococcus, Pelagibacter, Senecacoccus, Rosiobacters, and so on, that are very common in a lot of marine environments. So I think that this information that we are going to see here, while coming from coral reef environments, is possibly very applicable to other environments too, yet to be seen, of course. So we found over 3,000 individual phage host interactions in these samples. And when we de-replicate them at the species level, and we de-replicate the different sites that we visited 18 sites, we had 97 species level phage bacteria interactions. And this is just a visual representation of these interactions. The circles are the hosts, the triangles are the different bacteria, and then the different types of lines are the different types of interactions. So interactions that we believe are representing lytic infections or lysogenic infections, those where the viruses are integrated. We also have a lot of viruses that carry metabolic genes that have the capacity to change the metabolism of the host. Unfortunately, we're not going to have time to go over that here today. But so from this visual representation, of course, we can start pulling out a lot more information. And what we see here in this figure on the right side is simply the abundance of different bacterial groups in these samples. So you have that pink group at the top, those are your cyanobacteria, so they are grouped by phylum. And on the left side of the plot, you have the frequency of phage host links. And the first thing that you see that is very striking is very few links with cyanobacteria. And on the other hand, an overrepresentation of links in some other bacterial groups that are not very abundant. So you have the gamma protobacteria with a lot of links relative to their overall abundance in the data set. But you also have an overrepresentation of SAR-324, which are delta protobacteria. You have an overrepresentation of Planktomycetia and some other groups. So this was already indicating to us that there wasn't very straightforward relationship between abundance and frequency of links, which was very interesting to see. And of course, we had to investigate further that low frequency of links of cyanobacteria, which I can discuss with whoever is interested in cyanobacteria. But bottom line is that this has also been seen by other groups using completely independent methods, like Debbie Lindell's group with the pollinating method and Curtis Sorrell's group with the mortality by ribosomal lysis. And so it is a picture that is emerging in the field that, for the most part, cyanobacteria, even though there are a lot of viruses classified as cyanophages out there, cyanobacterial lysis, for the most part, is not that frequent. Okay, so then we start seeing this disconnect for a lot of groups between abundance and frequency of links, which makes us ask, so how do these viral links relate to the viral and the host abundances? And can we explain the frequency of these links by the abundance? Is this just mass action? There's more lysis when viruses find their host more frequently, right? So to answer to that question, for every water sample that we got, we not only did the high C sequencing that I just talked to you about. Oh, actually, okay. But we also got cell-associated metagenomes and free viral metagenomes. So these are the viruses that are associated with cells in a 0.2 micron filter. So these we will interpret for the most part as viruses that are actively replicating, or they are integrated in the cells, or they are associated with cells in some way, as opposed to viruses that are free in the environment. And we're going to get their relative abundances from these two types of data. And then, sorry about the animation. It's really not working well here, but we're going to get there. And then we also coupled with these microscopy-based total cell and viral abundance in these environments. So this is very important here because up to now, we were limited to relative abundances, right? The problem with relative abundances, to answer this type of question, is that if you compare two samples and the relative abundance of a virus goes up and the other goes down, you don't really know if it's because one grew or because of the other one disappeared. And so when you have something like your 100%, right, your total, your absolute abundance of virus and bacteria, then you can normalize your relative abundance and get finally the number of bacteria and viral genomes per milliliter of sample. So that's what we did here. We combined that with our pairs to get our abundance resolved phage bacterial network. And this is how it looks like. So just to walk you through this plot, we have the abundance in genomes per ml of either the bacteria or the viruses in the y-axis, and they're just sorted by their abundance in the x-axis. So you have the rank on the left side, you have the most abundant ones, and the right side, you have the least abundant ones. The top curve is representing the viral genomes, and then the lower curve, the bacterial genomes, and then you have the different types of links. You have Hi-C links that are interpreted here as putative lytic infections, and then you have the prophage links, which are those temperate infections. The first thing you notice, of course, is the lack of links up there on the upper left side of the plot. And then again, a lot of cyanobacteria there that were they just didn't have any links, right? So just as we saw before. And we also saw a lot of highly abundant viruses without links. That was surprising, and it's really not a product of the Hi-C method. So we did a lot of different analysis to rule that out. Basically, the Hi-C has a bias that is favoring finding links among those highly abundant bacteria and viruses. So the fact that we're not finding them is really striking. And one of the things that we thought about for explaining that high abundance of viruses without links is that they might have extremely long decay rates. They might be extremely stable in the environment. They might accumulate, even though they are not actively undergoing infection, at least during this time frame that we sampled across these 18 sites. Another explanation is the possibility that these are very recent infections where the virus killed the bacterial community as a whole in a very synchronized lytic event. If that was the case, when we looked at our 18 sites that we sampled across 2 weeks, you would expect to have a high standard deviation of the ranks of these viruses, right? So they would go up and down in rank because of this synchronized lytic event. And that was not what we saw. So right here we have the standard deviation of ranks at the top and the standard deviation of abundance at the bottom. And even though we do see a high standard deviation of abundance in that top left side of the plot, the ranks remain very similar across the sites. So it was still very hard for us to try to infer what was going on with these bacteria and viruses that did not have links. So for the next analysis, we're going to focus on the ones that do have links and then try to infer what is going on there. So this is what I'm showing you here. We have the abundance of the hosts in the x-axis and the abundance of the viruses in the y. Each dot is a pair. So we have 17,000 plus pairs here. So these are all those 97 pairs, but in all the 18 sites. And we saw a significant but weak relationship between the abundance of the host and the abundance of the virus. And the black dots are depicting the viruses that are free living, and the gray dots are the viruses that are associated with the cells, so most likely inside the cells. And even though the relationship was a little bit stronger for the ones that are cell associated, this weak relationship was striking because when you look at the bulk community, when you're not pulling specific viruses and specific hosts, this is what you see. You see a much cleaner relationship between abundance and bacteria and viruses in the environment. So this decoupling between abundance and abundance of viruses and hosts was really striking to us. And so we try to go toward viral genomes per ml, as I mentioned. This did not improve the relationship very much. And we decided to use the ratio of viral genome copies per host genome copies as a proxy for productivity here. So, and I'm using the word productivity as an indication of anything like large burst sizes or differences in infection time or large fractions of host infected, but basically anything that will create many more viruses per host, then I'm going to call this viruses more productive. And then we see that there was, again, a significant relationship with viral abundance, but a very weak one. So then to make sense of that, we created this sort of framework where we are relating here in the x-axis this ratio of virus to host, but in the cell-associated fraction and in the free viral fraction. And then when we sort them by, the upper and lower quartiles of each one of these metrics, we end up with these 4 categories of viruses on the upper left with what we infer as long decay, so they stay in the environment for very long, or viruses with just very high production in the upper right, and viruses with low production, likely most lysogeny in the lower left. When we plot the data, then now we do see a much stronger relationship between these 2 metrics, the cell-associated and free virus to host ratio. We can observe, again, a much stronger relationship with the host abundance. So and that relationship is negative. So the most abundant hosts are in the bottom left of this plot. These viruses are infecting different hosts. So the colors here, I'm not going to get into the details of the taxonomy here, but then that means that we can start pulling apart these relationships between viral life history traits and the host that these viruses infect, which may be helpful for models. So it does agree with the frequency of prophages, where we have a lot more of these temperate infections in the lower left of the plot than you have in the upper right. So I think that I opened a lot more questions about what life history traits of viruses explain their abundances. But in summary, we found this relationship between the productivity and the abundance of hosts that might help us start understanding which viruses are doing what in these environments. So I think I'm a little bit over time. I'm sorry about that. And thank you for your attention.
Thank you so much for the talk. That was excellent. I think we're going to hold questions for Cynthia until the panel discussion later and move on to our next speaker because we're moving on in time. So I'd like to introduce Cristina Mojica, joins us from the University of Southern Mississippi. She'll be talking about using techniques to actually quantify the viral shunt, so viral-mediated lysis and carbon into the dissolved phase in the North Atlantic Ocean.
Okay, well, I am so excited about viruses being included in this session. As you'll see, I'm very passionate about including viruses in biogeochemical models. And so basically, my research looks at the interaction between microbes, both grazers and viruses, with their host or prey of phytoplankton and bacteria, and how those interactions are forced by environmental factors and influence the flow of carbon in marine ecosystems. Most of my work focuses on field-based measurements in the North Atlantic, and over the last 14 years, I've had the pleasure of sampling about 168 stations in the North Atlantic. So I'll be sharing some of this data with you today. So Jordan and Kate did a really nice introduction of this, so I won't spend too much time on this. The carbon in the ocean is basically a balance between your phytoplankton here that are fixing inorganic carbon and moving that carbon up the traditional classical food web or sinking out a euphotic zone. And then on the other side, over here on the right, we have the bacteria that are scavenging that carbon that's lost to the dissolve phase and reincorporating that into bacterial biomass, which can then be respired back to the atmosphere or taken up by bacterivores and reintroduced into the higher trophic levels. And so this is kind of the traditional thought and how we model carbon in the ecosystem at the moment. However, this picture is far from complete because as we kind of heard, viruses are there and they're doing lots of things. And so what I look at is this lytic cycle that's kind of been mentioned a couple of times. And so lytic phages infect both phytoplankton and bacteria. And as you've seen, the ultimate result of this is the bursting of these cells to release the viral progeny. And in result, it is also releasing all this organic matter into the ecosystem. And so in this way, this is called the viral shunt. These viruses are taking this carbon from away from higher trophic levels and shunting that material into this dissolved organic matter pool, which can then enhance bacterial remineralization. However, direct estimates for this activity of this viral shunt are really rare, and consequently, we know very little about this impact on biogeochemical cycles. So this is where I come in. So I look at all these rate-based measurements. So I look at production and also standing stock concentrations and also the partitioning of mortality amongst grazers and that viral lysis pathways. And I also do it over here for the bacteria. And so this is kind of what I'll be presenting today. So I don't think I need to tell anybody in this room why I do all this work in the North Atlantic. The North Atlantic is like a particular hotspot for productivity, and most of this productivity occurs every year in the spring. And because of this, this spring bloom has been studied for over a century. And what this figure shows is that the annual pattern in phytoplankton biomass, which is shown here in the black symbols, varies according to PAR, which is shown in the red, and changes in mixed layer depth. And so accordingly, even though during the early days of Hawk and Bran, which is over 100 years ago, mortality was recognized as an important process dictating the accumulation of phytoplankton biomass, because of this pattern and because of the way that we were because of our lack of ability to reconcile these accumulation rates using our traditional grazing experiments, it has really come to the forefront that phytoplankton blooms are caused by abiotic factors such as shallowing mixed layers, increasing light, and associated with nutrients. However, we now know that viruses and microzooplankton grazers are important mortality factors for phytoplankton communities, and they are key players in these carbon and biogeochemical cycles. However, they are seldomly and explicitly parameterized in ecological and predictive models. Typically, they are included as a linear and density-dependent mortality term. And so the data I'll be presenting today, I hope will convince you that these general loss terms are insufficient to capture the patterns in energy and accumulation rates that we see in nature. So I'm going to start out here with two cruises and a stratified oligotrophic North Atlantic. Here we have Stratopete and here we have Medea. And so really we're looking at a situation where we have strong density stratification and nutrient limitation, which kind of weakens here in the northernmost region. although the entire transect was still classified as being strongly stratified. And so looking at total loss, that being viral lysis and microzooplankton grazing, against division rate here on the x-axis, you can see that total loss was tightly coupled with division rate, indicating a very high turnover in biomass. And more importantly, it shows that these two loss processes together were sufficient to balance phytoplankton division rate and reconcile the net accumulation rates, the low net accumulation rates we see in these stratified oligotrophic regions. And this is consistent across all different phytoplankton groups measured. And then if we take that total loss data and separate it into our viruses, which are shown here in the red bars, and our grazing shown in the blue bars, over our different phytoplankton groups, you can see that phytoplankton mortality rates pretty much evenly distributed amongst viral lysis and grazing pathways in the stratified region. And this says a lot about the flow of carbon in this ecosystem. And what's more, if I then take those rates and look at the ratio of viral lysis to grazing right here, shown on the y-axis, over that latitudinal gradient, you can see that these are not randomly distributed and that there's a pattern here. And what you see is in this southernmost region, where we had that oligotrophic, highly stratified conditions, we have a dominance of viral lysis, meaning that those points are falling above that dotted line. And when you get to these higher latitudes, where we have higher turbulence, we saw that there was a real big switch towards a grazing-dominated system. And this was, again, consistent across all my different phytoplankton groups. And what we found is that this relationship was actually driven by a decline in viral lysis rate that was negatively correlated to turbulence. And this is important, and this will be re-emphasized throughout my talk. So now if we move to another transverse across the Atlantic during the spring, we have a different situation where we're in spring, so this is April, and we had basically all these stations in this red box here being classified as non-stratified. And so again, if I show you that same plot of looking at division rate versus loss rate, you'll now see that it's not a one-to-one relationship, that you actually have more excess division relative to loss. And if you look at that a different way, when we look at the accumulation rates, we can see that we have positive accumulation rates across our different phytoplankton, which is really what you expect to see in spring. So again, measuring these two mortality rates really allows us to reconcile accumulation rates in phytoplankton. And so again, if we partition that across our different grazing and viral lysis pathways, you see a much different situation in spring than we did in summer. Reminding you, in summer, they were pretty much equal across all phytoplankton groups. And here you see that there is a dominance of grazing. Again, this pattern was found that the virus to grazing ratio decreased with stratification of the system. So viruses seem to be negatively impacted by turbulence. Also during this cruise, we examined the implications of altering the light environment experienced by the phytoplankton. So essentially, we were looking at what happens when you take a phytoplankton population from a deeply mixed surface layer and incubate them under static, shallow conditions. In other words, we looked at the implications of changing light environment on the balance between division and loss. And so essentially, we have our in situ conditions, and then we incubated them at the 15-meter water depth. And what you see is that you see this response in division rate to changes in PAR that is really mimicking the photoacclimation response, as you see here by the model by Platt et al. Being applied to that data. Another important thing is that due to our short incubation times of 24 hours, we were able to decouple our losses from our phytoplankton growth rates because of the slight lag in the response of our mortality factors to catch up to these increases in growth, because we did not see a significant effect of light on phytoplankton mortality rates. So the result is now shown here, and what it's shown is that accumulation rate is proportional to our delta mu, or the specific rate of change in growth. And this is suggesting that instead of phytoplankton accumulation being a consequence of abiotic factors regulating growth rate, it's really the decoupling between growth and division. And this provides clear experimental evidence for the disturbance recovery hypothesis. And if you guys are not familiar with this hypothesis, it's basically looking at phytoplankton blooms from the view of physical processes that decouple growth from losses and result in changes in phytoplankton accumulation rates. And this is important because if we start to think about this and how we model phytoplankton blooms in our models, this is going to have a much different impact on how our phytoplankton communities respond to climate change, right? It has actually the complete opposite sign as what we currently consider. So as stratification increases, we're going to have less decoupling between our growth and loss, meaning we're going to have a negative impact on our phytoplankton communities. So I've really been focusing on this side, and so I'm going to go ahead and switch over to this side where I'm looking at bacteria. I'm running out of time, so I want to go pretty quick. So let's start out with the summer stratified conditions again. And here I'm looking at viral-mediated mortality versus protist-mediated mortality. And what you see is that viruses were by far the dominant mortality factor, and they were responsible for an average of 55% of the total mortality, which we've kind of already heard this morning is within what we kind of know. Another thing I was looking at here is there are two distinct populations that you can distinguish in your bacteria using flow cytometry. They're called high nucleic acid and low nucleic acid fluorescent bacteria. And that's because this fluorescence property is linearly related to the genomic material within those populations. And so I was curious on whether these HNA LNAs were important factors regulating my viral dynamics. So here I'm going to show you this, and I know everybody hates RDAs, so I'm just going to run you through them. Even us ecologists do not like them. But basically, it's a multivariate analysis that lets me look at many different types of data at once and test hypotheses about what factors are important in driving my distributions of my data. And so basically, I'm just looking at the first two dimensions. You have one dimension for every variable included in your model. And the first two dimensions usually typically explain most of the variability within your data. I have my explanatory variables shown in blue here and my response variables shown in red. Only variables that were significant to my model, as tested by Permanova, are included here. And basically all I want you to see is that this HNAs, the length of this line also indicates the significance. So these HNA LNA population or ratios were very important not only to my viruses, but also to my grazers. The second thing I want to point out is that viral activity and grazing activity were governed by distinct physical-chemical forcing. For viruses, we have host availability, encounter rate, and this ratio of these two different groups. And conversely, here for my heterotrophic nanoflagellates and my grazing rate, it was really temperature. I also want to point out the importance of turbulence to both of these groups. And again, we see this negative correlation between viral production and turbulence. We saw that in phytoplankton, and now we're seeing that in bacteria as well. What's more is we saw this kind of inverse relationship between our heterotrophic nanoflagellates and our virus abundance that were positive correlated in the mixed layer, and then in the lower depths we saw that this kind of fell apart and we actually had a negative correlation, meaning that these are negatively influencing each other. And what you see is that there's a difference in the abundance of our host or our prey items in this mid-depth. And so it seems that there is a negative interaction between protists and viruses when host or prey abundance is low. And this could both be for competition for hosts, but also because protists often can graze on viruses. So when they don't have their preferred prey, they can sustain themselves on viruses. Finally, here I'm looking at total mediated mortality over total available carbon, and total available carbon being standing stock carbon plus production. And what you see here is that we move from a steady state situation where you have a high turnover rate of this bacterial biomass, and then as you move out of this oligotrophic zone, you really move to being a net heterotrophic production. Another thing that you have to consider in the North Atlantic is that it's an energetic eddy field. So how are these eddies influencing these rates? Again, I'm going to go really fast because I have like less than 5 minutes. But basically, we went out there and we sampled all these different eddies, inside, outside, anticyclones, cyclones, and also outside. And I looked again at this relationship between HNAs and LNAs. And this is because, just really quick, LNAs are believed to be oligotrophic, and HNAs be copotrophic. And so this is kind of verifying that. We saw that this HNA LNA ratio increased with abundance as well as production per cell, suggesting that indeed that HNAs take up more carbon than LNAs. When I did that RDA analysis, again, I'm not going to show you that because I don't want to bore you, but phytoplankton, carbon, DOC, and eddy field were significant in explaining the abundance and composition of my heterotrophic bacteria community. So the environment is driving my carbon availability, which drives my bacteria community, but what about the viral community? Really what I found was that, again, it was this ratio was important, the abundance of my host, the growth of my host, and also over here, we have the virus to host ratio. And what I want to show you here is that we really saw that we had this HNA LNA being related to my V1 population, both in abundance and production. So we have a similar stack of viruses when we look at them on the flow cytometer. And basically, these lower ones here were more correlated with these HNAs. And so if I look at this more closely and I look at viral V1 lytic production over HNA abundance, we see this really nice relationship. And what you see is that now these populations that we can distinguish on the flow cytometer are giving us some kind of ecological information, and this more productive virus is correlated with a more productive host. Didn't get to the end shot, but basically what I can do is I can take all this data and put it in a model and show that the viral shunt is way more than we thought it was. Previous predictions are 25%, and my data shows it's between 33 and 80%, where I have coupled rates in the stratified region. Thank you.
All right, thank you. So next up, we have Sherry Floji from Wake Forest University. She'll be talking to us about beyond viral lysis, understanding the role of infected cells in marine planktonic communities.
Awesome. Okay. If at some point I start to trail off and you can't hear me, just like wave your hand around or something. It's really hard for me to stay close to the podium because when I teach, I tend to like go back across the whole room, and so this is a challenge for me. So don't be afraid to let me know if you can't hear. Okay. So as marine microbial ecologists, we know that we face this problem of understanding that microbes are interacting at levels of single cells, right? So we have microbial interactions are occurring at single cell levels, but we want to understand ecosystem or global scale processes. So connecting these single cell interactions with global scale processes is a grand challenge. And so we're going to switch scales a little bit from ecosystem and regional scales down to single cell interactions. And I'm going to talk with you about a project we've been working on for a few years, trying to understand how viruses impact the chemical composition of infected cells, what those chemicals are that are released from infected cells, and how those chemicals might mediate interactions with neighboring microbes. And so really focusing on cells before they lyse, before they have this really dynamic and obvious bursting phase. This was a multidisciplinary project. Can you guys all still hear me? We're good. Okay, awesome. Okay, so this is a multidisciplinary project bringing together marine virologists, microscale physicists, and chemical ecologists. And let's see, again, as we were saying, we're trying to identify the chemical compounds that were released from those cells and how they mediate interactions. And I just want to start by thanking OCB, one, for letting us have this session, but also for the opportunity to share this work with you. And also the NSF Biological Oceanography Program funded this work. Okay, so as we know, complex planktonic food webs control biogeochemical cycles. Roughly 50% of global photosynthesis is occurring in the oceans, primarily carried out by microbes, and 1/4 of photosynthetically reduced carbon cycles through this really dynamic pool of seawater metabolites. And these dissolved organic carbon compounds support growth of other organisms, as we've been talking about, but they also serve as information sources. And so they're cues to other microbes and larger organisms. Viruses are central in these ecosystems and in these food webs, but we have a hard time quantifying the role of these, and emerging techniques are coming out to try to better understand this. Viral lysis is something that we historically, as a field, marine virologists has focused on because it's easier to study and it's easier to work with cells that show this really obvious phenotype of dying when they're infected with a cell. And so this is an electron micrograph of a bacterial cell that's being lysed by bacteriophage. And when these cells lyse, they create a short-lived chemical hotspot. This is a graph showing the size of that hotspot, basically the size of the phytoplankton cell radius, and then how long that chemical pulse would remain in the environment before being diluted out to background chemical background levels. And so, as you probably know intuitively, larger cells will create a larger hotspot that will last longer than smaller cells. And so, historically, phytoplankton have been thought to not generate enough of a hotspot to really drive many microbial interactions. Phycospheres, on the other hand, are long-lived. So these are long-lived chemical hotspots. A phycosphere is just a three-dimensional dissolved organic matter matrix that surrounds a cell. Okay. As you heard about from Kate and some others, viruses manipulate host metabolism. So during infection, they are expressing their own genes. They're also manipulating the gene expression of the host, and they're changing the biochemical content of the cell and also the chemicals that are released. And that could potentially alter phycosphere composition. So phycospheres historically have been considered to be static, but we're starting to understand that the phycosphere itself can be dynamic. And this is a plot here, which wasn't included in the viral primer. And so this is showing the number of extracellular phage particles or virus particles over time in an infection. So when we add viruses to our cultures, there's a little bit there. They go down in abundance as they absorb to the host cell. And then there's this stretch of time where it seems like nothing is happening. The host cells remain high, the virus particles are not seen, but really this is where all the biology is happening, right? So the virus is taking over the host cell, it's manipulating gene expression, it's changing the composition of that cell. And then we start to see virus particles being released into the extracellular phase. We often also think of the burst of viruses as being this big synchronous event, but it occurs over time, even within a clonal culture, phytoplankton culture or bacterial culture, with a clonal virus added. And so some cells will burst earlier and some cells will burst later, and that burst size can vary as well. And then ultimately we get to a phase where all of the cells in that population that were going to lyse have lysed. Often that's not 100% of the population, even in a clonal culture. And then we have the total number of viruses we'll receive during that infection. And this is a schematic of what's happening. So this has been studied quite a lot in marine picocyanobacteria. And what you're looking at is a schematic of a cyanobacterial cell where the red arrows indicate the different pathways that are manipulated by viruses. And so Cynthia mentioned auxiliary metabolic genes. So some of those in cyanobacteria can manipulate the Calvin cycle, so shutting down the Calvin cycle, manipulating energy and flow into the pentose phosphate pathway. We can increase nitrogen uptake, increase phosphorus uptake in these infected cells. We can deviate resources towards more pigment biosynthesis, and this can change the susceptibility of an infected cell to photoinhibition, so basically decreasing photoinhibition of an infected cell. And then ultimately, there's a lot of deviation of resources towards nucleic acid synthesis and protein production for the purpose of generating viral particles, so viral DNA replication. And so what we did was a study where we had a controlled infection looking at Synechococcus, and we took samples for transcriptomics and metabolomics over a time course of the infection, sampling pre-lysis and post-lysis. And what we found is that there are a number of metabolites that are enriched in the media in virus-infected cultures relative to uninfected cultures. And these metabolites are things that are really labile, like nucleotides, amino acids, vitamins, and all sorts of other resources. And it got us to thinking, especially because some of this metabolite release was prior to cell lysis, how could these metabolites be influencing interactions with other organisms? And so put it another way, we say, do virus-infected cyanobacteria release chemoattractants that can stimulate microbial interactions? And so a chemoattractant is just any chemical that can be sensed by an organism that will then modify its motility pattern. So a chemosensing bacteria can basically sense a chemical gradient and then change its motility to either swim toward or away from that compound. So positive chemotaxis is swimming towards something, negative is swimming away. Okay, and then we broke this down into sub-questions. The first is, are virus-infected Synechococcus releasing compounds that are different from uninfected cells? We already had some evidence of this. This was conducted in my lab primarily by my grad student, Jonathan Moon. What are these compounds? And this was carried out by Trent Northern's group at Berkeley Lab. Benjamin Bowen and Suzanne Castina worked on these samples and data. Do these compounds influence mortality or induce motility in chemotoxic heterotrophic bacteria? And this work was spearheaded by Jeff Goasta's lab in the mechanical engineering department at Tufts University. And then last, is the release from infected cells sufficient to generate a chemotoxic response and or enhance interactions? So we ran a highly controlled infection experiment with Synechococcus WH8102 and the virus SSM5. We infected the cells, we let the viruses absorb to the cell, and then importantly, we then washed the cells and removed the free virus and resuspended in new media. And then we collected samples for cell abundance, extracellular phage abundance, and then exudates for both metabolomics and chemotaxis assays. And I just want to highlight your attention to the gray box. So this is the latent phase, or the phase where the cell density is not changing and extracellular viral production is not changing. And then this is highlighted here. So up until 4 hours, we had no change in cell density, and up until 6 hours, no change in extracellular phage abundance. And this is what we found in the comparison of exudates from infected and uninfected cells. We have enrichment in certain metabolites shown in orange, and also a depletion in certain metabolites. And some of those are interesting. So Suzanne Craig yesterday was mentioning some production of industrial type compounds that's produced by organisms. And so we see some evidence of these things that we had to go back to our data and really confirm that this wasn't contamination from our methods. And then these are the plots of the peak height of these specific metabolites over time. So blue is control and orange is infected. So methylthioadenosine was a big one, as well as deoxyadenosine. Particular metabolites, and then also phenylalanine and palmitamide. And so our next phase was to measure bacterial chemotaxis to the exudates from infected and uninfected cells, and then measure bacterial chemotaxis to select compounds that we had identified. And the way that we do this is this is a single assay chemotaxis device. You can flow infiltrates from uninfected phytoplankton on one side, from infected phytoplankton on the other side, and then you put your test organism in the middle. And what we do is we quantify the number of cells in the top part or the left part and the bottom part. And we characterize the chemotaxis by this factor called beta T, which is the number of organisms on one side minus the number of organisms on another, divided by the total number of organisms on both sides at the final time point. And what this looks like is this. So you can see the bacterial population is moving toward one side of the channel over time. We see this sharp increase in beta T. And then over time, as that chemical gradient relaxes, the the bacteria start to explore the rest of the channel. And so I'm showing you this because I'm going to show you a whole bunch of plots that look like this one on the right. So hopefully this will make sense to you now. Okay. Short stories, we found that Vibrio alganolyticus exhibited a preference for exudates from virus-infected Synechococcus. And so each of these plots is showing beta T, and they're each from a specific time point in the infection, beginning at the beginning of the infection. The top three plots are before the cells were lysine, and each of the colors is a different biological replicate, so a different flask that was infected. You can see there's some variability there, but a lot of agreement. In our Ret B shown there, we actually had a slower infection, so we have some biological explanation for why that one looks a little bit different. And then during lysis, we also see that there's sort of a decrease in beta T during the middle of infection, and we see the highest positive chemotaxis towards infected exudates early in infection and late in infection, post-lysis. Okay, we also repeated these experiments with Pseudomonas haloplanctus, and we just did 2 time points, so the 2 hours post-infection and 12 hours post-infection. And what we found is Pseudomonas is only attracted to the exudates that are being released early in infection. And so you can have differences in chemotaxis with different organisms. And I wanted to highlight for this crowd, especially after a lot of the talks yesterday, is that not only do we have a complex mixture of metabolites in these exudates, but each of these chemical compounds will have a different diffusion coefficient. And so some compounds can diffuse very quickly far from the cells, like VOCs, shown in yellow, and others have sort of a medium diffusion, that like amino acids, and then others, like polysaccharides, will stay close to the cell for a longer amount of time. And it's also important to note that the chemical compound that serves as the cue to stimulate chemotaxis is not necessarily the same compound that the organism takes up or assimilates. So it can use a different compound to cue it in to a cell or to a nutrient hotspot. Okay, so at this point, we wanted to test model compounds that we had identified in our exudates, but we ran into a scaling problem, which was that if we wanted to test 4 different organisms, have 5 different gradients plus a control, and run 5 biological replicates, suddenly we're up to 120, and this is just for 1 chemical compound. And so there's a graduate student in Jeff's lab who developed a multiplex chemotaxis device. This is the schematic of what the actual design is, but the beauty of this device is that it conducts on-chip dilution. So you don't have to manually make the dilutions and then run all of those separate assays. And so it dilutes tenfold with each channel. And so you can simultaneously run 6 different chemotaxis assays. You just program your microscope stage to move beyond. So you're basically sequentially repeating through those different channels. And this is what the single assay channel looks like on the bottom left, and the multiplex channel. And this is Michael. He got his dissertation. He's now a postdoc at Brandeis University. Okay, so now we were equipped to move forward with using this MCD to quickly quantify chemotaxis towards multiple compounds. We first started with our model stimuli. So serine is a model chemoattractant, and phenol is a model chemorepellent. So we can see that we're having a positive chemotaxis towards serine, negative towards phenol, which is a good control. And then we added a bunch of our compounds that we had discovered or identified in our infected cell metabolomics data. So there are a couple of things I want to point out here. One is that we observed both positive and negative chemotaxis through these different metabolites. and that the concentration stimulating responses varied by compound. So for leucine on the bottom, it was only at the highest concentration, so 200 micromolar addition, that we start to see positive chemotaxis, whereas for deoxyadenosine, it's quite a bit lower, two to 3 orders of magnitude lower, that we're seeing a response. Okay, and then these are each of those plots, but I'm not going to go through them, but these are all posted on bioRxiv if you're interested in looking. Okay, so our last phase was, okay, so we have these exudates. We ran these culture experiments at pretty high density. It's about, let's see, I think 10 to the 5 cells per mil. And so what we wanted to do was run the experiments at a lower density and just see, with Synechococcus is at a more environmentally relevant density, can bacteria sense them and swim toward them? And so we ran another experiment where we infected cells, we then washed them, resuspended them, and then we put the cells themselves into the chemotaxis channel, and we put the test bacteria in the middle, and then artificial seawater was on the other side. And what we saw again is that, yes, we see positive chemotaxis towards infected cells. And notably, we didn't see any chemotaxis towards uninfected cells. And that's that black line there. And then that's at 3 hours, so it's pre-lysis. And then post-lysis, we see an even higher chemotaxis towards those infected cells. I will note that the cells that we put into the channel, there was no change in abundance of those cells. There's about 20 per channel, and they were roughly 100 microns apart. And so we don't think that we had enough cells in the channel to have overlapping phycospheres or like a big buildup of exudates in that. And so, yeah, we see chemotaxis towards those at both time points. Okay, so I'm just going to wrap up by talking a little bit about how we think about phycospheres and how we think about microbial interactions and how viruses fit into all of that. So typically when you think about phycosphere-mediated interactions, we think of big phytoplankton, right? So diatoms and other organisms that can generate these big radii of DOM. And the reason for that is that the length scale, the run scale of a bacteria before it turns and switches directions is less than the radius of the DOM pool that's produced in a phycosphere by a big phytoplankton, so by a diatom. When you start to get to smaller phytoplankton, that phycosphere is much smaller, notably. It was thought that bacteria couldn't hone in on these small phycospheres, and part of that is because much of the work was based upon our knowledge of E. coli. E. coli thrives in nutrient-rich environments, and so there's some more recent work that's come out in the past couple of years looking at marine bacteria. It turns out marine bacteria actually, they're calling them high-performance chemotaxers. So they're just more attuned to low levels of DOM and able to respond to them better. And while they may not attach to the cells, and they may not stay within the phycosphere as a single cell, they are able to enhance their time spent around the cells in these phycospheres. Okay, and I'm going to skip past this because I'm running out of time. So the big question is, what is the impact of virus-mediated chemotaxis? And then these are the conclusions. So we know that diverse metabolites are enriched in the exudates and the phycosphere of infected picocyanobacteria, including vitamins and compounds rich in carbon, nitrogen, and sulfur. The chemoattractants are released at all stages of infection. During early infection and post-lysis, heterotrophic bacteria can sense and respond to these compounds, no response to infected cells. And microfluidics provides a scalable platform. So if you're interested in this, I encourage you to come talk to me, check out the eLife publication, and we have another publication with more detailed protocols on the way.
Okay, thank you to all of our speakers for staying on time. We're actually a little bit ahead of schedule, so I think we get a slightly extended. Sure, we can do that. So we'll go ahead and do some questions for the first 3 speakers. We're going to have a panel discussion with everybody later, but this gives you a chance to ask questions while speakers are still fresh in your minds. So why don't we have the speakers come forward for the morning? And Amelia, are you in the audience? Yes. My microphone assistant here.
I have Okay. Hi, this is Hanji Wang from University of Rhode Island. Thank you for the talk. I learned so many things today. It's eye-opening. So as a chemical stenographer, maybe it's a very silly question for you all. So like, seems like the virus infection is very important on like hourly time scale. So if we study the daily time scale or monthly things, can we assume, given the abundance of this virus in the natural ecosystem, can we assume that it's in a steady state? Can we use like one coefficient to represent this in a local coastal environment or open ocean? Thank you.
Hi, Christina Mojica for people online. I don't know if you can see me. So I think the ocean is dynamic. And so what that showed is like in a steady state situation, like a stratified oligotrophic ocean, things are pretty much in steady state. But any change in that, change in mixed layer, a storm rolls through, so that is going to change. And you're going to be missing that response from the microbial community. So I don't think that you can use one coefficient for everything. I think that's kind of what we're doing now. And so the evidence I was trying to show is that's not sufficient to explain the patterns that we see in nature. Yeah.
Hi, Tatiana Reinerson, University of Rhode Island. Sherry, I really enjoyed your talk. Thinking about what the bacterial cells are doing in response to pre-infected or early infection. And I'm curious, you saw this response at the very end. You were talking about cells chemotaxing towards infected cells and not towards uninfected cells. What do you think the motivation is? Are the bacteria getting to those infected cells so that they are there when the cells lyse, or are they getting there because they're taking advantage of some metabolite that the cell is already exuding that they could be taking up in some dissolved form?
Yeah, that's a great question. So the next sort of follow-on experiments to these is looking at actual resource exchange, so stable isotope labeling the different organisms and seeing that exchange, and does it change over time? So what's the impact of nutrient or resource transfer pre-lysis versus post-lysis? And we see that there are both things that could potentially be deterrents as well as things that can be attractants that are released. These two organisms, the Vibrio and the Pseudoalteromonas, exhibited positive chemotaxis toward them, but it's possible that other organisms would not be swimming toward them. And then the other sort of big next step for this work is looking at the protist responses. I have a graduate student working on that now. So are some of these chemical cues that are released pre and lysis actually inducing or helping protists to find infected cells? Do they then eat them? Are they assimilating those resources? Or is something else happening? Thanks.
Hi, I have two questions. The first is, does the chemical environment affect the future response of the viruses when you're doing your chemotaxis work? So if you expose them to one level, will they continually respond to that level, or do they kind of become saturated with it and don't respond? And you need a higher level.
For the bacterial cells?
For the viruses to go to the bacterial cell.
So the viruses are typically infecting the cells based on contact.
Just contact.
Yeah, so I don't believe the viruses are responding to any sort of chemical cue, so that's more at the bacteria level. So okay. How are the bacteria responding to chemical cues induced or released by viruses? We can start to like wildly speculate, viruses are manipulating the cells to release cues that then maybe bring other organisms close to them that they can then infect, or, help them aggregate more so that they can be closer to other host cells. But I'm going to just pause and say that's complete speculation.
And the second question is UV. Does that affect viral populations?
I'm sorry, can you say that again?
Does UV, ultraviolet radiation, is light important to viral populations?
Yeah, absolutely. So UV will will facilitate degradation of free viral particles. So a virus is safer inside of a cell. And then light itself is important, particularly in infections of cyanobacteria, but potentially in other bacteria as well, or other hosts as well, where you need light energy to facilitate a productive infection. And there's lots of data showing that.
Okay, we've got time for 2 more questions. I've got one online, and Susanna is going to take the other one, and then we'll save the rest for the panel.
Do you want me to go first?
Sure.
Oh, hi, Susanna Neuer, Arizona State University. I actually have 2 questions. I hope that counts. So the infected, so I was wondering about loss of viruses themselves, right? Can they be grazed? I mean, obviously they have a lot of juicy DNA or RNA. Are they attaching to aggregates? Or do they even make cells more sticky that are, you know, what Sherry presented, maybe that's also a stickiness thing? Can those cells maybe be more easily integrated in aggregates that sink and export? Maybe I'll leave it at that question for now.
So the short answer is yes to all of the above. They can be inactivated by particles. They can be associated with TEP and also removed from the environment. These things have all been shown. And they can be grazed by protists, which there's a lot of nice studies coming out that show that that's actually pretty important in the environment. And so I showed some field data that also suggests that that could be important when a host abundance is low. So yes.
Okay, one more question from George Capellos online. Thank you for the great talk. I'm curious whether the high chemotactic precision of marine bacteria is due to chemotaxis alone or also due to chemokinesis, modulation of speed.
Oh, yeah, that is a great question. I don't know the answer to it. I think that there's a lot of work to be done understanding chemotaxis of marine bacteria and bacteria that are like isolated recently from the marine environment, rather than working with these, model organisms that we've worked with for a long time that have been cultured for a long time, and also quantifying their behavior.
All right, we're going to go to break, but before you get your coffee, please go down. When you go out the door, please go to the left, exit the tent, and go down towards the water. We're going to grab a quick group photo with T-shirts. So if the faster you go, the longer you get for coffee. Thank you.
Welcome back, everybody. Glad to see you here for our sort of next phase of virology session, which is thinking about viruses in models. And so David Talmy is going to speak with us next. So empirically modeling carbon flow through marine viruses and microzooplankton grazers. And he can come on. He joins us from the University of Tennessee, Knoxville.
Cool. Thank you all for having me here today. So the overarching sort of motivation of this session and my talk is to try and understand how interactions among viruses and grazers influence the flow of material in marine systems. So here I'm showing phytoplankton as the prey or the hosts, but we can also think about many of these processes influencing bacteria as prey or hosts. So there's been some discussion about whether viruses influence the flow of material towards sinking, removal from the surface layer, sometimes referred to as a viral shuttle, or perhaps toward regeneration of material on the surface, more traditionally thought of as a viral the shunt, or towards higher trophic levels. The approach I'm going to take is really a modeling one, and it's kind of in 3 parts. The presentation is in 3 parts. So I'm going to begin with a case study in the California current ecosystem, where we try to model carbon flow through viruses and grazers. And then I'm going to talk about host virus life history traits, try and extend to more diverse organisms, potentially to larger spatial scales. And then show some early work where we try and model viruses on some larger scales in the ocean, and emphasize some of the biomarkers that can be used, that have been touched on in some of this morning's presentation, to help sort of constrain and interpret model predictions. So starting with this first part, this is a case study from the California current ecosystem. So the data that I'm going to show are kind of similar to some of the data that we've already seen this morning. So this was a sample site, or 4 sample sites that's shown here in this chlorophyll map. Number 2 is a little bit hard to see, but there are 4, and they go across a gradient in chlorophyll. And at each of these sites, Alexis Basolka and colleagues performed dilution experiments, which I think is where we were getting a lot of the data that we looked at this morning, that Katrina and Jordan and Kate showed. So the way this works is there's a dilution factor on the x-axis. So this is the amount of seawater. I should say I've never done one of these experiments, so everything I'm saying could be completely wrong. So there's a dilution factor on the x-axis, and that's the amount of seawater that was filtered through either a 0.1 micron filter, which gets rid of the grazers but leaves the viruses, or a 30 kilodalton filter that gets rid of the grazers and the viruses. And so then you look at the change in chlorophyll over time to get an apparent growth rate. The fact that both lines are negative is because there's more sort of predators or grazers, viruses, where there's less dilution, and the difference in the slope of the lines can be used to infer a virus versus a grazer loss rate. So taking those values inferred from those dilution experiments and plotting loss rates on the y-axis as a function of the chlorophyll concentration on the x-axis here, we get this interesting kind of relationship with chlorophyll concentrations just from these 4 sample sites, but it's kind of intriguing, right? And so these are just rates, and so we want to go from here to movement of material to a flux. And so we did that with a model. The model more or less kind of reproduces what the data seems to imply in terms of the relationship between mortality and, in our case, phytoplankton carbon, but it differentiates between the viral biomass, the grazer biomass. What I'm calling the viral shunts are just the leftover stuff, and then sloppy feeding and respiration, so what the grazers do with it, okay? We can't differentiate between DOM and POM and thinking versus regeneration, but this is perhaps a step in the right direction. So I'm going to get into some of the weeds of the detail of how we made this model. So I think this might be the simplest kind of way that I could think of to couple phytoplankton, viruses, and grazers. So if we have a very, very simple model, which is just a homogeneous pool of phytoplankton and a homogeneous pool of grazers and viruses, there's a phytoplankton kind of mass balance here where the phytoplankton grow at rate mu, but are infected by viruses and lost to grazers following the simple mass action Holling type 1 kind of relationship. They then get transferred up to the viruses and the zooplankton with some transfer efficiency, epsilon V and epsilon Z. And so I'm going to talk about how we can use dilution experiments to quantify these 2 parameters, phi V and phi Z, and then also think about the transfer efficiencies, epsilon V and epsilon Z. Okay, so this is kind of the result that we get when we use the dilution experiments to get clearance rates. Essentially what we do is we take the mortality rate and we kind of work backwards and divide by a biomass or an abundance of the viruses and the grazers. And we can, if we have kind of a quota model, we can put it all together and get these estimates of a clearance rate, right? So this is kind of my bread and butter as a modeler. Like if I want to write down a model, this is about the most basic information that I would need. The nice thing about these dilution experiments was that they measured the viral abundance and the grazer biomass, so we could go from mortality rates to get these parameters out. The fact that the virus clearance rate is higher than the zooplankton clearance rate is something that I'm going to come back to in part 2. Okay, so I've done the clearance rates. We're going to move on now to the transfer efficiencies. This is difficult to measure directly, but if we know the virus carbon quota, if we have some sort of even a model of the virus carbon quota, we know how many viruses or the burst size are produced after infection, and we know a host carbon quota, then we can infer a transfer efficiency by putting these two things together in this quite simple relationship that we can compare with known transfer efficiencies for grazers. So grazers tend to get somewhere around 10 to 30% of their host carbon. Viruses, it's much lower. This is using a compilation of bacteriophage and viruses of eukaryotes. So much, much lower, and I'm plotting as a function of size here, right? So the viruses are smaller than the grazers. So when we put this together, the transfer efficiency is what gives that very thin slither. The viruses get a very small portion of their host's biomass. That's why that red slither is very thin. The blue, solid blue wedge is a bit thicker. And then there's all this waste material, right, which could have a different composition, but this model doesn't differentiate. Okay. So this is one study site. There are more dilution data. Christina showed some other dilution data this morning. But we really want to go to like really large spatial scales. And so because the parameters that I got that I showed, particularly the 5V and the 5Z, are specific to that study site, it's difficult to use that in a model because there can be all kinds of changes in the composition of the viruses as you go to other systems. So we want to try and extend to larger spatial scales. And so the trait approach has kind of been the most successful at doing this. So I want to talk about how host virus life history traits can potentially be embedded within this sort of a framework, or this sort of framework can be extended to incorporate the trait approach. Okay, so, and by the way, I apologize, I'm mixing up all kinds of language here, right? So I wanted to try and use a common language. So I'm calling everything a predator. We heard viruses being talked about as being parasites this morning. That's probably more appropriate. I'm sorry if I upset anyone. It's just easier to find one word to describe both viruses and grazers. So like anything that has to encounter food, that has to consume some resource, we have to first encounter it, capture it, and then transfer some of that material. You can think about a very simple model for a grazer that swims. Not all grazers do this, of course, but many do, to find its prey. Viruses, on the other hand, are dependent upon Brownian motions. They move around in this kind of random fashion. And then the grazers will ingest some of that food, and there's some waste product, either respiration or some exudates. But then the viruses will make more copies of themselves, but release this lysate, okay? So can we characterize these three different stages? For encounter, it turns out there's some very, very old biophysical models that we just kind of put together in some of these review papers. They're not really review papers, but sort of synthesis papers. So shown here is a theoretical encounter rate for a motile predator. That's the blue line. So the bigger you are, in theory, the wider your clearance rate. Not clearance rate, the wider your detection radius is, and the faster you can swim. So you get this increase in encounter, the bigger you are. But if you're a predator that depends on Brownian motion, the bigger you are, the less influenced you are by molecules moving you around. So that goes down. Interestingly, the vertical line happens to be roughly the size of the largest known viruses and also the smallest motile predators. So this is just theory, but we can compare this against virus absorption rates and grazer maximum clearance rates, and there's some correspondence there. The ribbons just account for differences in the host size. In theory, this should set an upper limit. The encounter, the ribbons should set an upper limit on the capture, which it sort of does, and there's kind of qualitative agreement here to suggest there's some biophysical control on capture. We want to then go from here to this third stage, which is transfer. Virologists talk a lot about burst sizes, right? And that makes a lot of sense because, you know, higher burst, potentially higher proliferation. I struggle to think about that in a kind of a carbon context because a very small virus could have a very large burst size, but how that plays out in kind of carbon units can be confusing. So to sort of go through this whole stage, I'm going to convert the first two to be carbon specific by just dividing by a quota for the virus and the carbon, for the virus and the grazer. And that kind of changes the relationship that we get as a function of size. So here it looks like Brownian motion is very, very good relative to swimming, just generally speaking, because we've divided by this small viral carbon quota, okay? And then that also works for the capture. Carbon-specific capture is quite high for these viruses. This is just for lab cultures, so not natural communities. And then if you then extend this also to account for transfer, this suggests that maybe viruses have a higher encounter and capture, but maybe they have a lower transfer efficiency, right? So maybe this kind of levels out in some way in the ocean, and possibly explains why you get some of these data suggest sort of matching or equitable sort of losses to viruses and grazers. Okay, and just a note, going back to part one, we got this number 5e was higher. This is comparable to what I'm calling a clearance rate here, or maximum absorption. There's just 4 sample sites from this one study, so I don't know if this is really true. But the idea, that the theory at least predicts that the viruses would have a higher carbon-specific clearance rate. So we probably would need more of these kinds of data to try and know if that's really true generally. Okay, so now I'm going to talk about some early work trying to extend to some larger scales in the ocean. So we took some of these kind of biophysical, empirical compilations and developed a version of the Darwin ecosystem model where we had 2 different size classes of phytoplankton feeding on a pool of nutrients and grazed upon and also infected by a bunch of different viruses. This is not nearly as diverse, right, as some of the data we've seen this morning. That's kind of just a note that we've only got 5 viruses here. The waste products of viral infection and grazing go into POM and DOM, and we had to make some assumptions about how that partitioning happens. And then that gets fed upon by different classes of bacteria, and then that in turn kind of feeds back into this POM and DOM pool, so it's kind of a mess. But we can put this into the MIT GCM and just solve it using the existing kind of trait framework that the Darwin ecosystem model uses. And this is what we get. Okay, so this is a kind of first order prediction. We're thinking of these as being lytic viruses. On the lower right here, we've got phytoplankton in the upper left, bacteria upper right, zooplankton. So this is aggregating among those different types. So it's perhaps what you might expect, you know, where you have sort of higher phytoplankton abundance, the rest of the ecosystem kind of follows along, kind of like a rising tide lifts all boats. And that comes from the modeling, which is basically just assuming kind of a mass action type interaction. But then the question is, well, okay, we've got these predictions. How do we interpret them? How do we know if any of this really makes any sense? I think there has been an explosion in the development of sort of techniques to try and measure and monitor viral infection, some of which we've seen today, some of which was in some of the webinars if people are interested in the run-up to this meeting. I'm going to give just one example, but you know, I'd love to talk to people who have more of these kinds of data. So this is some cruise data from the North Atlantic. There was a cruise led by Kay Beidel and team. And so they were monitoring coccolithophore bloom and these Emiliani huxley viruses that infect the coccolithophore bloom. And here is showing genes that mapped to an enzyme, serine palmitoyl transferase, that I'm not going to talk about in detail, but the key point is the colored bars can be mapped to lab isolates that we know information about their traits. Okay, so EHB86, 207, and 99B1, they have different phenotypic properties. 86 and 99B1 are relatively slow to infect their host. 207 are fast. And these lab experiments kind of show this. So here we've got cells infected at T0 by a type of fast and slow infecting the same host. And so the blue versus the red is this kind of slow dynamic that's happening the blue relative to the red. The model is on the upper right here. It's a bit more complicated than the model I showed before, but by solving this relatively simple model, we can kind of capture the difference between the slow and the fast. And we can then take this trait information. What's nice about this is, you know, the phi, the beta, if you make some assumptions about the quotas, you can get to the exact same framework that I showed in parts 1 and 2. But we can begin to play games. We can ask, what if we compete this slow versus the fast virus in Darwin? And then ask different hypotheses. So what if the fast virus is infected or is removed at a faster rate? And there is some motivation for this. There could be more clumping because they release more sort of sticky particles, and there was a higher number of reads in some of the deep ocean measurements. And then what if there's a more diverse host population that the slow and the fast virus can infect? We can then put these different scenarios into Darwin and ask what happens. The left column is the area roughly corresponding to that cruise tract that I showed, and the vertical dashed line is just the point where we show the depth profile on the bottom row. And so if we just put the lab kinetics for these groups together, we get that the fast virus just always wins. There isn't coexistence. But if we have this differential loss or this different host subpopulation, we can get coexistence, which is more consistent with the environmental data. Okay. So in summary, I think that the host virus life history traits are a really important way to extend to larger spatial scales. These kinds of games with model interpretation and different ecological, playing around with different ecological scenarios are enabled by these different biomarkers. I showed genetic biomarkers, but we've got lipid biomarkers, transcriptional biomarkers, quite a lot of different types of data that we can throw at this. I should just give a plug that Steph Giekewitz has a poster talking about some of the viral V-Darwin. And next up, Daniel is going to be talking about some stuff extending this not just to carbon, but nitrogen and phosphorus. There's lots of, I think, kind of ongoing questions, so partitioning between DOM and POM. I know there are some groups who are working on this kind of thing, so Kim Tomatricone and the viral folks and the Safadi and others are thinking about how we can partition viral waste between these different pools. We heard a little bit this morning about lysogeny, these kinds of long-term infections. Okay, this is really messy from a modeling point of view. Mathematically, it's really hard. And we need to think, I think, as a community, is how we can sort of resolve that. And then, of course, this diversity, right? So the models, we can't capture the true diversity that we have in nature. And so how we sort of parameterize that, how we account for that is a kind of an open question. So I think I'm coming in just under time. I've said questions, but I think we're not doing questions until the end. Thank you very much.
So please hold your questions for the panel, which will be just after Daniel's talk.
Okay, so howdy y'all. My name is Daniel. I'm extremely excited to be here. This is my first OCB, so thank you again so much for the invitation to come and speak today. The lineup of talks that we've had has been incredibly exciting, and we've had some questions that I think get us to this place at I'm a biogeochemical modeler. Viruses are super vast and diverse. I'm scared. Hopefully my talk will attempt to allay some of those anxieties and get us to a type of functional trait-based framework for viruses like we do for diverse ocean microbes. I'll also throw out that this work is partly supported as being one of the inaugural cohort of the BioGeoscape's Early Career Fellows Program. So if you're interested in that, I would be happy to talk about it as a sort of like a cousin of OCB, I guess. And then because I'm a completionist and they requested that we cite Vicodemo data, this is all based entirely off of theory, but I have deposited data to Vicodemo before, so also a great resource. All right, so this morning we talked about the viral shunt. And the important thing here is that through lytic infection, the matter from the host is partitioned between the viral progeny that results from the infection, as well as the dissolved organic matter or detritus that gets released back into the environment. And as David just explained, because viruses are very small for the most part, the cellular mass ends up in the detrital pool, which David described as a low carbon transfer efficiency. And so if we want to try and extrapolate that to the global ocean and understand a little bit more detail about the biogeochemical implications of this like very intense shunting, one strategy is to pick a host virus pair that we already know a lot about, like David described with EHUX, and then implement that in a global model. That's what this team here, the V-Darwin team, is attempting to do with including viruses in the Darwin ecosystem model at the full global scale. And if you're interested in more details about that, then I would recommend checking out Steph's poster at the poster session. But my job in that group is to try and think about how we can generalize to hosts and virus pairs that we don't necessarily know a lot about, or host virus pairs that might be represented in a model that don't have a perfect analog to a microorganism that we have a lot of microbiological knowledge of. And so one way to start to get at this, and the way that's implemented in Darwin, is using size as a master trait and organizing other functional traits of microorganisms based off of their size. This has a whole bunch of different advantages where we don't have to super strongly condition every organism we include on our model on a whole bunch of difficult to gather and highly uncertain physiological data. Instead, we can use physical principles such as diffusion and maximum possible swimming speed, like David alluded to in the last talk, to set the traits of these organisms based off of their relative sizes to each other. And then the other advantage of this is that we can use size distribution data to help us inform how well our model outputs are doing, and size distributions also have a relationship to bio-optical data that we can observe at large scale. So how can we start to systematize viral traits in the same kind of way? So using some pretty simple geometric and first principle arguments, we can start by talking about how does the macromolecular content of a virus vary with its size, the relative ratios of different macromolecular pools. Then if we make some assumptions about the relative elemental contents of those macromolecular pools, we can extend that to get at viral stoichiometry as a function of the virus size. And then from there, we can think about the host size and the virus size in the same transfer efficiency framework to think about the composition elementally that we would expect for viral lysate. And so a very oversimplified physics geometric-based view of how viruses work is you can essentially think of them as a bunch of nucleic acids in a sack of protein. And so that's what this a paper from Luis Jover in 2014 used as a basis for making these scaling arguments about viral particles. The idea here is that you have basically spherical viral capsid that is a protein shell that is its surface area, and then the volume is filled with nucleic acids. And then from there, if you make some assumptions about the carbon, nitrogen, and phosphorus content of protein and nucleic acids, then you can start to account for elemental totals of viruses of different sizes. And then from there, get to stoichiometries. So this was the state of the art 10 years ago. We're trying to add a little bit more sophistication to this model now that we have much more data about viral physiology and more environmental sequencing of viruses. So the first step here is to think about, is the bag of protein always completely full of nucleic acids? And when we look at the data, it turns out that actually no, as viruses get bigger, they get relatively less full of nucleic acids. And in fact, it depends on the virus architecture, what types of biomolecules it's made out of. So here I'm comparing bacteriophages, which are the viruses that we mostly heard about this morning, that tend to be very full of nucleic acids. But when we get into the really large giant viruses, like the EHUX viruses that David talked about in the last talk, they get relatively less full. And then we can also think about other macromolecular considerations. So enveloped versus non-enveloped viruses, the giant viruses like the EHUX virus, in addition to having the protein sac, that's also coated in a lipid membrane that is derived from the host membrane phospholipids. And so we can make a similar scaling argument here about saying for as much as the protein sac is then inside of a lipid sac. And so from there, we can get to updated allometric scaling of viral macromolecular constituents and incorporate the differences between the relative amount of DNA fill and the outer structure, macromolecular structure between these two different sort of big, broad virus classes. So that lets us get a little bit at the stoichiometry of the overall viral particle if we make some assumptions about the CNP content of the lipid envelope, the protein shell, and the nucleic acids. looking in this bacteriophage constant genome cell original model, you can see in general, the larger a virus gets, the closer its stoichiometry resembles just pure DNA. But in general, that viruses tend to be much more nitrogen rich and also much more phosphorus rich than cells, especially red field. So with this new approach, we can kind of expand on that idea a little bit and fill out some of the gaps in the picture. So this is looking as a function of either the virus size, or I've included some small cells here to give you a point of reference. What we expect the carbon to nitrogen, nitrogen to phosphorus ratios to be. Notably, carbon to nitrogen is systematically lower in viruses, but in bacteriophages is much lower because they don't necessarily have the lipid envelope. And then the nitrogen to phosphorus ratios are also much lower than cells. Also because bacteriophages are so much nucleic acid or proportionally. As they get bigger, they actually get more nucleic acid rich, and so that's what causes the NDP ratio to decline with size as opposed to cells where the NDP ratio generally slightly increases with size. So now that we have this allometric scaling and we can sort of have a slightly more detailed recounting of the elemental quotas of viruses of different sizes, then we can start to play some games about trophic transfer in different elemental currencies. So the data set that I'm going to be talking about here comes from this paper using TEM imaging of single infections from a lake in Japan. I like microscopy-based approaches for playing with these games because we can simply look at the images and then count the number of viruses to estimate birth size directly, and then we can also directly measure the virus sizes and the host sizes, which we can use to calculate these quotas in the transfer efficiency framework that David proposed in the last talk. And so this is just to give you a little bit of a picture of what those three parameters look like. The other kind of thing that I'll hint at here, but we're still developing, is the relative sizes of the hosts and the viruses may also encode additional useful information about their functional traits, such as the average birth size. And then for getting these host and virus volumes into elemental totals, I talked through the allometry on the virus side. If you're interested in the allometry on the host side, that's what the poster that I have in the poster session is about. So you can ask me about that later. So we can calculate all of those transfer efficiencies based off of those quotas and the measured birth sizes. And then, in general, the carbon transfer efficiencies that we calculate for these single infections track with what David's model estimates. That carbon transfer efficiency on average is very low. In fact, in this data set, we estimate the average carbon transfer efficiency is only about 2%, which means 98% of the carbon from the infected cell ends up in the detrital pool. And then we can move on to other elements. And then because of the stoichiometric differences between viruses and their hosts due to the different macromolecular architectures, we end up with substantially higher nitrogen transfer efficiencies than we do carbon transfer efficiencies. So you can see the y-axis here, instead of going up to about 10%, goes to almost perfect transfer efficiency. Then again, we can move on to the phosphorus transfer efficiency. And that's even higher, where in fact some of these we would estimate that the virus would require more phosphorus than might already be in the cell at the time of the initiation of the infection. And so like Sherry was mentioning earlier, viruses can encode auxiliary metabolic genes that enhance uptake of different resources. Phosphorus stress-related genes are the most common of these auxiliary metabolic genes. And so it might have to do with this increased phosphorus requirement to build all of these nucleic acids to get the viruses assembled. We can also look at how these transfer efficiencies compare to each other relatively. So when we look at that framework, we can basically look at how many times more efficient is nitrogen transfer than carbon transfer, for example. And in this data set, we can estimate that across all of the infections that we see, nitrogen transfer is at least 10 times more efficient than carbon transfer, which means that the material that's getting sent into the viral shunt to be recycled is much more carbon rich than would be expected from a non-virus mediated sort of general mortality process. Yeah, the other thing that I'll point out here is that if you have a large virus that has fewer progeny, you end up with better nitrogen transfer than if you have a tiny virus that has a large burst size. We can play the same game for nitrogen to phosphorus to show that the phosphorus transfer efficiency is much higher still than the nitrogen transfer efficiency. And so that's like in Sherry's talk, how the metabolome from the infected exudates are rich in carbon-heavy and nitrogen-heavy compounds relative to the cell. That's also what these allometric scaling models would predict. Then the reason that this line is the different shape is because the virus nitrogen to phosphorus ratio and the host phosphorus nitrogen to phosphorus ratio actually scale in opposite directions of each other. Similarly, you would rather be a small virus with a large burst size for less total phosphorus transfer. Yeah, so sort of landing on the importance of viruses for carbon cycling and biogeochemical modeling, the big take-homes here are based off of like pretty simple geometric arguments and a little bit of real-world data to help constrain them. We can estimate the macromolecular composition of viruses as they infect their host, depending on what type of virus and what type of host they are. We can transfer that to elemental totals and look at differences in stoichiometry between viruses and cellular life, and then implement that to understand that this viral shunt is not only like a remineralization enhancing process, but also is an elementally disproportionating process. And so the main thing that if you come home with any of this, you can think about the viral shunt actually enriches the detrital pool in carbon relative to other elements, while those other elements are retained in the viral progeny. And then I'll basically leave it there. As we head into the discussion section, other topics that might come up that might be of interest, the stoichiometry that I referred to actually is subject to natural selection and has an evolutionary biogeography that you can feel free to ask me about. Viruses and metals also have a lot to do with each other, and is a very interesting story that we could talk about. And we can also talk about, like in the talks this morning, how ocean physics can sort of modulate the relative intensity of the viral shunt and can constrain it to small spatial environments. Yeah, so I'll leave it there.
Okay, I think we're scheduled to move straight into our panel discussion. So if all the speakers want to come up and sit at the table here.
Okay, so Sherry is going to put up our guiding questions for this section just to sort of get ideas flowing, but this is free form, so feel free to fire away at the panel. And remember to say your name and your institution, and Amelia and I will run the microphone to you.
Hi, thank you all for a really nice set of talks. I'm Bethany Jenkins from the University of Rhode Island. And my first question really goes to Daniel. I really like the framework he's set up for thinking about this. And I'm wondering, is there evidence to show that there's less viral lysis in P-limited ecosystems? Is there a constraint on these P-limitation, you know, I'm thinking about the Sargasso or a classic P-limited system. And then the converse of that is, do you see anywhere where high levels of viral lysis actually can drive a system further into phosphorus limitation?
Thank you so much for the question. For the relative intensity of the viral shunt, I might leave that to you, Kristen, to take a stab at. But yeah, I will say I have done a little bit of field work in the Sargasso looking at the distribution of viral infection for cyanobacteria, and we have some pretty strong evidence of a good seasonal cycle in where, like, shortly after restratification, signatures of viral infection are at their highest, also slightly beneath the mixed layer. And I think that ultimately will stimulate primary production because of the recycling due to the viral shunt. Yeah, and potentially, I don't know, also maybe like the extent or the time scale by which the phosphorus stays sequestered in the viral pool, I'm not entirely sure. I think it would be very interesting to look into that in a little bit more detail, for sure.
What was the question about the intensity of the viral shunt? No, I haven't really seen this. And another thing that was shown in the lab experiments is that they can use organic bound phosphorus. And so that might release some of those limitations.
Hi, Corday Selden, Rutgers. So this idea of elemental disproportionation through the viral shunt is really interesting and obviously has important implications for carbon sequestration potential. So this is kind of a broad question for the group. Do we have a good sense of what drives the relative importance of the viral shunt within an ecosystem? Like, where does it matter and where does it not matter in terms of carbon cycling?
So we see that in stratified system, it's super important, right? It's the dominating mortality factor. And this actually is very interesting because it can help sustain this high productivity that we actually see, right? It's a high turnover, and it's likely this release of these nutrients and organic matter that's driving the system. So.
Hi, Julie Granger, UConn. Regarding the transfer efficiency, this question may be of interest to many in the room, but what would you guess the transfer efficiency of iron would be?
What a fabulous question. So for folks who may not be familiar, a couple of years ago, Chelsea Bonet and Maya Breitbart's group posed this thing that they call the Ferrogen horse hypothesis, which based off of 3D structural assessments of a virus of E. coli that finds a small amount of iron atoms in the virus tail. It suggests that the viruses actually might have a small iron quota. It was just a handful of atoms. But in super iron-limited marine systems, what they pose in that paper is that siderophore uptake receptors might also actually be the viral uptake receptors in those cases. And so that means the viruses basically get all of the advantage of the super high uptake affinity for iron to be very efficient at infection. I have a little bit of ongoing work looking at estimating metal cofactors in viral proteins, working with Sarah Walker's lab and her postdoc Felipe Benitez at Arizona State University. So we'll be able to have a slightly better quantitative understanding of what virus iron requirements might be. But we do know that iron that gets released in lysate is highly labile, and probably is also bound in all of the various humics and goodies that are inside the cell anyway. And that iron limitation and viral infection pressure might actually be sort of like contravailing forces in the system coevolution. So you might end up with suboptimal iron uptake due to the enhanced risk of iron infection. It poses a very interesting evolutionary game theoretic problem.
Hi, Greg Cutter, Old Dominion University. So I've always been interested in the history of science or oceanography. Have any of you done the plots since, let's say, started in 1980 of number of viral publications versus time? Do we see a sinusoidal behavior of that? And my other observation is this is usually driven by technological changes? I'm old enough, by the way, to remember all these.
I can say that I have not done that as an exercise, although it would be interesting. One thing I've noticed is that there was a lot of interest early on when we realized viruses were abundant and active of the role of viruses in biogeochemical cycling. So there's some very well-cited papers trying to model the impacts of viruses in biogeochemical cycles and nitrogen and carbon flux. And then over time, there seemed to be a dip in interest in part due to the technological challenges. I think the impetus for this session is that we're reaching a time where we're starting to have a lot of new tools to tackle this problem.
So make a plot. It's always good for an introduction. I have another ignorant chemical oceanographer question. When I see something that's so well distributed, the abundance of the viruses that you had, it suggests to me a long residence time, that they're well mixed. Can you tell me I'm crazy?
I agree. I think that viruses do have a long residence time. So I think UV decay really is only really important in that very upper layer. And like you mentioned that they have this, some like Seneca has this diol infection cycle, so they have strategies to avoid that. And when we look at rates of decay due to binding to organic matter or sediments, these are really low compared to the production rates.
Mortality. There we go. It seems that it's based off of sort of size fractionation characteristics in looking at the free-living viruses, but what about the ones that are already in the cells? Does that mean that you're actually underestimating viral mortality in the dilution experiments?
Yes, this is one of the limitations of the dilution experiment. So one thing is I don't look at bulk chlorophyll or total populations. I look at the individual populations that I can distinguish on flow cytometry. That's how I get those different populations. And so we found that really matters in being able to distinguish these different rates. So the second part of your question was, yeah, so I'll let you follow up. So the assumption of the method is that it's based on infection that's occurring within that 24-hour period. That's when you dilute out the mortality pressure, you'll see this increase in growth. But that is one of the limitations, is that there could be infection cycles that take longer than that 24-hour period that you're missing, and also that there could be ones infected already prior to. And so I know that David has looked at this.
Yeah, I think Christina knows about the dilution experiment way more than I do. Latent period has to be shorter than the duration of the incubation, right? And then you kind of get enough sort of replacement happening during the experiment for it not to be such a huge issue, but for the longer, exactly as Christina, I'm just saying what Christina said. But there are other kind of bottle effects you can get, and so Stephen Beckett and Joshua White had some studies, some sort of modeling studies showing how sort of hypothesizing that if you had like an R strategist and a K strategist, and as you do the dilution, could get different phenotypes in the host, right? So there's various ways that the dilution technique can give results that you might have to sort of correct for in some way.
Hello, Luai from Hui. I don't know much about viruses, but since they're so abundant in the ocean, I can't help think of them as like little delicious packets of food. So how important is like organisms that eat viruses in that whole model system that you guys are talking about? Or should that even be considered viral? Yeah.
So as with a lot of this, there's a lot of sometimes conflicting data on this. So like there's some older experiments. So the buzzword for that is virovory, I think, and I challenge you to say it 7 times really quickly. But there's some older experiments with some viruses where that was not thought to be a significant thing. David Demery had a nice PNAS paper just recently where they sort of thought it was in the Corella virus. A much more significant movement of carbon, right? But the grazers can be kind of picky about what they eat. So it's kind of
I was just going to point to the same papers. So now there's recent studies that are coming out and it's showing that this could be something that we should take into account. But one thing about that is, as you can see, that they're not really loaded in nutrients. So it's only under a situation where they're starving, right? And they don't have anything better to eat.
Julie Granger again. Okay, this is a little early, I guess, in the game because these aren't in the models yet, but how does that translate to the transfer of carbon relative to nitrogen and phosphorus to the deep ocean?
Yeah, so this is what David and Steph and all of our colleagues and I are hopefully going to find out. But there's sort of two primary considerations to me. So there's the relative transfer efficiency process. So the detritus is like enriched in carbon relative to nitrogen and phosphorus. Whether or not that detritus is partitioned between DOM and PUM and the relative aggregation dynamics and sinking speeds of that detritus is still an enormous question. There's also been anecdotal data that suggests that for like diatoms in particular, infected diatoms have a regulatory response to induced sinking. So it's sort of like a noble sacrifice to prevent other diatoms from becoming infected later, right? So there's still a lot to be parsed out about how viral infection, while it's happening, and then also the properties of viral lysate and the viral progeny themselves end up in sinking and export. There's also a question as to how sticky the lysate is relative to how sticky the virus progeny are, because you also could make the argument that the progeny viruses, of course, and then the lysed cell are very close to each other in space. So if they're both sticky, they might mostly all immediately aggregate together, and then it doesn't really matter. So that's something that I hope we can make some more progress on figuring out.
One thing one of us should have said was that from the Tara Oceans studies, there's some older papers, right, where, I think I'm blanking on the name of the first author. Is it really? I'm probably going to say it wrong, but they found that I think it was Synechococcus phage, right, was the best predictor of carbon export. And so I don't think that we have enough understanding to know whether that's because they're driving carbon export or because they're just most associated with events that are involved with carbon export in some way. And everything that Daniel said is important in helping to try and pass that out.
Let me just interject really quick. Cynthia, we have you up with the panel at the front of the room. So if you want to jump in at any point, just raise your hand and we will give you your chance.
Yeah, and another really important consideration about that result, though, is that how they estimated carbon export was based off of like list data, and so it's more closely interpreted as Synechococcus phage are associated with large particle size. But there have been other metagenomic studies that like Elaine Law, who's now at UNC, has done that identify cyanophage like genes in abyssal depth sediment trap particles. So it's like we know that they're getting down there, but it's still very much a big question as to whether or not it's actually more export or not.
Tatiana Reinierson, University of Rhode Island. So I just want to say what a fun and interesting session this is. And I wanted to thank Kate and Jordan for their really interesting overview talk. I wanted to follow on some of the things you brought up in that intro talk, and one of them was the real diversity of viruses that are out there, and you really did a great job of pointing that out. Cynthia, in her talk, said, well, 95% of the viruses we can identify, I think is how she said it, I'm not sure, we can identify are the DNA bacteriophages. So can you help me tease this apart as I walk away? Should I just be like, really, it's all the DNA bacteriophages, or how much do I have to really be thinking about the full diversity of the viruses that are out there?
Well, I can start answering that one. So there is debate, right, on what is it that we are seeing. Is it really the real representation of the diversity and abundance, or is it a bias of methods, right? So it's really much easier to identify double-stranded DNA viruses. You can count them in a flow cytometer or in an epifluorescence microscope much more easily than you can do with an RNA virus or even a single-stranded DNA virus. And some of the methods that we use for detecting these other groups of viruses, they tend to bias heavily toward them. So when you combine these different methods for detection of the different types of viruses, it's really tricky to get a good quantitative picture. But I think that even if this number that I gave you was lower of the representation of the double-stranded DNA bacteriophages, it would not be lower, like an order of magnitude lower for certain. I think that all the methods that we have point to a dominance of that group, except of course when you have, for instance, these really big blooms of eukaryotic phytoplankton that is followed by a bloom of eukaryotic viruses, right? So then you have a dominance of these eukaryotic viruses. And in terms of viruses that infect bacteria, the single-stranded and RNA ones, or the single-stranded DNA ones and the RNA ones, I don't think we have seen any evidence that they would surpass the double-stranded DNA viruses in abundance, even if they are more abundant than we are estimating right now. So I don't know if other members of the panel want to add to that.
I was just going to say similar things, and the methods that we have really kind of target DNA, and that I've seen a few papers where they looked at using like qubit fluorometers to quantify RNA and DNA, and the suggestion there was that it's actually more of like a 50-50 split, but those aren't great methods to actually quantify that, right? So whether or not that's true is up for debate. And then the other thing to think about is also, what are we talking about open ocean? Are we talking about coastal environments? Something that's more human impacted, we might see more RNA viruses because a lot of those tend to infect like humans or animals.
Sorry, just add one more thing on that too. Can you ignore them? I don't think so, because when we talk about the dominance of one type of virus, it's because their hosts are the most dominant. But single-stranded RNA viruses are really important for diatoms, and RNA viruses are very important for protists. So it depends on who you're interested in, right? And can you ignore them? No, because we can't. Everything's interconnected, so we can't just ignore one mortality factor.
Hi, Charlie Coleman from Skidaway Institute of Oceanography. And my question is going to be kind of a Frankenstein question between yesterday and today. So go with me for a second. But I was thinking about Kim's talk where she showed the image of the phage in the aerosolized form. And I think a lot of the time we're always thinking about what the human impact is on the marine environment. And so I was just wondering, like, is there any research on aerosolized viruses and being deposited to open ocean environments, especially like viruses that are maybe more human derived and have maybe no business being in the open ocean, and how those, like, you know, I imagine you talked about photodegradation being a big factor, but if there's some potential for inorganic metals or something protecting the virus inside the matrix, but can these things be deposited there, and sort of just interested in that.
I think there is some evidence that they are deposited. One thing I would comment on is that viruses that are from humans or from freshwater systems don't really survive that well. So we talked about this long residence times of viruses. But viruses that are not used to the marine environment are usually deactivated, or they can't reproduce because the salinity has a lot to do with their ability to infect their hosts.
Go along with that, I think I've seen a couple of metagenomic studies where they were actually looking at aerosol viruses that were able to make it up into clouds and then transport it over long ranges. So it is an area that's starting to be looked at, but how much of that is actually happening is
Carly Buckwald, Dalhousie University. So my question kind of stems from the second question of the discussion starters. Why are viruses not included in ecosystem models? And I don't do ecosystem models, but I model nitrogen cycling processes. And I was thinking about in my models, like where do viruses come in? So for me, they come in, I have, ammonification, so PON to ammonium. And I'm thinking, okay, this is where the ammonium, this is where the viruses are helping make that PON go to ammonium. So now I'm thinking, sitting through this session, so why do you think I should care about how that PON gets to ammonium? Like, should I be considering whether it's through viruses or other types of death and mineralization?
I just want to make one comment. The reason why they're not incorporated, as you can see from the discussion, is it's complex, right? They're very diverse. They're very complex in their strategies and who they infect. And so this is the biggest limitation, I think, and I think that David and Daniel's talk really emphasize that. So, but I hope that we're making good progress in that direction.
Yeah, and also, I mean, I guess it depends a little bit on what your scope of interest is for the geochemical reaction work that you want to talk about, right? Because if you want to have a difference between like a sort of direct ammonification route inside of a cell versus release to dissolved organic phase first, then it's like, okay, well, that's when viral lysis would get you there. Also, like as I was suggesting, nitrogen is sort of differentially collected into viral fraction. And so, if you end up like with a buildup of DON that is otherwise not labile, because if you took it up, you would die, then, it's, yeah.
Yeah, I guess it is going to depend on like the scale that you're interested in, right? And if there's temporal spatial sensitivity of that pathway to viral infection, it's associated with the abiotic environment, then maybe you could probably parameterize it if you had enough data, but yeah, I don't know, you probably would do fine without it.
Hello, my name is Yishuan Song, like from University of Rhode Island. So I'm doing like physical oceanography, but I have an open question. Like if I'm doing a lab experiment and I have an assumption that like with the virus colonization or infection, the particles will getting more fragile like over time. So do you have any like suggestions or ideas? What should I do like for verifying this assumption? Like, how can I better control the experiment? So for example, if I'm testing the natural particles, but I really want some certain types of virus or something else, like, do you have any suggestions?
Yeah, I'm not sure I have suggestions. So there have been a couple of people asking about how quickly are viruses deactivated. And also going back to Greg's question about, we find virus particles all over the ocean, so are they very stable? So a particle is not the equivalent of an infectious virus. There can be lots of particles out there that can't initiate a successful infection. And so in our lab, we take care to generate a fresh virus stock and then work very quickly with it because, quite frankly, we don't want to go through the labor of quantifying that deactivation rate under different conditions in our lab. And so we would just work with it quickly in a time frame that we know that it remains highly infectious. And so would anybody else like to comment? Maybe it's a topic for discussion over lunch. Yeah.
Okay, John Lauderdale from MIT. I'm a kind of, not necessarily an ecosystem modeler, but definitely a biogeochemical modeler. The picture you're painting is kind of very complex, and I'm more of a impressionist, Van Gogh kind of guy. So to reduce it right down, can you give me the Frontier for Young Minds virus model, rather than the slightly more complicated, lots of arrows kind of model that David and Daniel are kind of illustrating?
Basically, I want to capture the feedbacks and the interactions, but with really reduced complexity. So, you know, can we at least make a kind of stab at that?
I thought we were showing the reduced complexity model.
Yeah, keep going.
If you wanted to be, well, like implicit viruses in ecosystem models now is just tack on one additional mortality term to all of your plankton, and then treat it exactly the same. That's like the least I think we can do outside of just completely ignoring them.
I would argue differently. So what you could see from the field data that I presented is it's very interesting that we see this negative correlation with turbulence, right? And so this gives us a way that we can now implement them in models, biogeochemical models that already have a turbulence term in there, right? And so viruses dominate when you have stratified conditions. And then you can know that you have a high recycling in those areas, but as soon as you get to a certain threshold of turbulence, it then partitions all that mortality to the grazing pathways. And we're seeing this consistent not just in the phytoplankton, but also in the bacteria. So I find this extremely interesting and also really exciting way that we can now incorporate viruses into biogeochemical models in a very simplistic and straightforward manner, because you saw that these patterns were consistent across all phytoplankton groups. So yeah, I think that that's a really exciting way for us to move forward. We still need to know the mechanisms and all the other important details, especially when it comes to the consequences for phytoplankton composition, for example. But when we're looking at the flow of carbon and now nutrients, which I think is really cool that you can use those parameters to say what happens afterwards to the carbon and to nutrients. But I think that this is a really great way for us to move forward on incorporating these.
Great. This is Nick Hako from University of Hawaii. So this is based off of something that Cynthia showed, which is the low infectivity of cyanobacteria, especially Prochlorococcus, which matches up with some data I've seen Station Aloha as well. And I guess, you know, the question I want to ask is, I've heard that it's not limited by contact rates, like the viruses are touching the cells, they're just not infecting it due to resistance and stuff. And so I guess I'm wondering, resistance seems like a really major consequence biogeochemically if it modifies phytoplankton traits. I guess I'm wondering across the group if you have ideas about ways that those aspects might be modeled as well as like the mortality processes that you've done too.
I think that that's really challenging, right? I mean, there's more and more being discovered about that biologically. And, you know, if we want to try and keep John happy, we just can't put all that complexity in there. I think that you can probably get some of these parameters for resistant versus susceptible types from the kind of lab experiments that we have, and then try and understand if those are selected in different environments. And then once you've got if you can establish a sort of meaningful environmental pattern, then maybe develop a simpler John-style model to parameterize it. But I agree. I think it's a really important and challenging question.
I think I can make a comment about this problem of the different levels of infection in different bacterial groups, right? I think that just like we had this question on should I care about incorporating viruses in the model if I'm looking at, let's say, some sort of nitrogen metabolism, I think that's going to be a question of who's participating on those processes, right? Are you talking about sinking? Are you talking about transferring to higher trophic groups? Are you talking about nitrogen metabolism? And then who in your bacterial plankton or phytoplankton community is mostly participating on that? And then is this group highly infected by viruses and frequently and at what rates, to define how we are going to incorporate the viruses and if we are going to incorporate the viruses, right? Because if it's if the bacterial group, for instance, that is doing that sort of nitrogen metabolism that you're interested in is not very frequently infected, is that really contributing to the overall picture? And if not, then you can probably ignore. We know, for instance, that in cyanobacteria, when they are infected, there is a really big shift in how the carbon is shunted inside the different metabolic pathways. But if only a very few cells are infected, for the most part, sometimes not. There are cases in which you have most of the cells infected. We don't know the environmental drivers for that, right? So I guess the bacterial group and the circumstance, the environmental context will define that.
Sorry, I'm just going to add one last thing to that. There's some great work out of Debbie Lindell's lab looking at, what the environmental factors are that might drive that high proportion of infected cyanobacteria versus non. And so I think that with some of these new methods, the Polleny method, we're moving toward a space where we can start to link environmental variables with percent infectivity, hopefully, even though it is complex. And then, as Christina was saying, we might get to a point fairly soon where we can link things that are easily measured by physical measurements to biology. Very exciting.
Hello, Yandash from University of Southern Mississippi. I have a question for Dr. Sherry. The chemoattractants that were released from the cell lysis of Synechococcus, can it be backtracked to how many cells were lysed, and can it be alternate measurements of the bird size variations that we always talk about in terms of production?
That is a great question. So we don't have quantitative data on the metabolite release per cell. And we were really focused on the release of certain metabolites pre-lysis and then the impact on the microbial community chemotaxis. There's work in the Miliana-Huxley system where they find specific metabolites that are associated with viral infection, and that can be used as an environmental tracer of blooms, of infection of blooms. We haven't found anything right now that looks like a smoking gun for cyanobacteria or Synechococcus in particular, but that would be pretty exciting if we could.
My turn. Okay, hi. Alessandro Taliaber, University of Liverpool. So I'm a biogeochemical modeler, maybe a bit more complex than John, but not at the same level as you guys. And so I think, yeah, on the one hand, maybe a density-dependent mortality, where we're sort of dealing with it. But I think the interesting arguments that make me think that we need to worry about it are the environmental dependencies. If there's a link to some environmental property that makes that mortality term more susceptible or more likely to be on or off, and then the biogeochemical arguments around the stoichiometry, that if we're, because if you density-dependent mortality, we just lose the cellular material in constant proportion to what's in the cell. So that, I think they're the two real lines of argument. But then I start to wonder about the size of these viruses, which, if I'm not mistaken, they operationally fit into the dissolved pool. So how much of the DOC, DON, DOP, or even dissolved iron is actually being modulated by viruses? If there's so many of these viruses there, this seems like a real headache for all of us, actually, modelers and observationalists, to understand these implications.
Yeah, so based off of the Hover paper that I was showing the schematics from in my talk, in terms of total DOC and total DON even is still pretty negligible, even though virus particle concentrations are very high. In low phosphorus regions like the Sargasso, the Hover paper argues that viruses could be as much as 10% of DOP. And then in the Bonane and Breitbart Trojan horse paper for the dissolved iron, they make an estimate. I don't want to misquote the estimate, but it's also pretty high, like of like FeL1 could be viruses, but that structural work still hasn't been finalized. We still aren't even entirely sure if the Trojan horse thing is actually happening. But because they're so tiny and not as abundant as just free individual molecules, so they still make a pretty small proportion of the totals.
Oh, hi, Kyle Niemeyer from Oregon State University. I think this is mostly for Christina. It was prompted by your discussion of turbulence. So I'm a mechanical engineer, I'm probably one of the few in the room, and come from the world of chemically reacting fluid flows. And one of the things that we do in that world is use certain non-dimensional quantities, non-dimensional numbers to try to collapse trends onto, from different conditions onto a same plot. And I was thinking of the Domkohler number, which is the ratio of turbulent mixing timescale to chemically reacting timescales. I'm wondering if that's a quantity that could be useful for you in characterizing some of the behaviors you see. It wouldn't be chemical reactions, but it could be the characteristic timescales of some of these viruses.
That's a bit above my pay grade. I have no idea what you're talking about. But we can talk about this in more detail. You can explain it to me, the frontier student version or whatever that is. It's an interesting thought, right? Because KT is temperature eddy diffusivity. So it's not just turbulence. It's that kind of chemical diffusion, which might be interesting. I'm also interested in the impact of Brownian motion on this. Yeah, I'd love to talk about it more later on a lower scale.
Sounds like you guys have a proposal to write. So it takes us right to 12:30. A reminder that we'll thank our panel.